Ok. Pardon. Ok. Je te le laisse par terre. Ou par là. Il est toujours allumé là.
Alors bah là je vais essayer d'éteindre mais je... Yes, perfect, yes.
So, hi everyone, thank you for showing up at 8:30 for the second part of the mechanobiology session. This morning we will have Nathalie Sauvonet and Matteo Raudi, and then later on Peter Lennart and Guillaume Charras, and we'll talk about the mini symposium also a little bit later. So, Feng, you can introduce Nathalie.
Morning everyone, so it's my great pleasure to introduce Nathalie for you. So Nathalie is a project leader now in Pasteur, leading a group of intracellular trafficking and tissue homeostasis. Nathalie did her PhD in microbiology actually in Pasteur, and afterwards she moved to University of Louvain, where she studied protein sorting in prokaryotes, and afterwards she switched to intracellular trafficking in eukaryotes in Pasteur Institute. So thank you, Nathalie.
Thank you very much. You hear me? Yeah. Okay, so thank you very much for the invitation. It's also my first MIFOBIO, so it's really great. I will come back. So today I will talk about the impact of physical force of a gut on pathogen infection using organ-on-chip technology. So you all know that each tissue in the body has its own particular three-dimensional organization with its particular interface, like liquid-liquid or even air-liquid interface, and they all encounter several physical forces, such as oxygen tension, but as well you have some stretching or compression or shearing force induced by the fluid flow. And all these physical parameters are very important for the tissue homeostasis and tissue function. However, this is not well known how these forces regulate exactly the tissue organization. And in particular, we absolutely don't know how these physical forces play a role during host-pathogen interaction. So today, I will focus on one organ, the intestine. So, as already Stéphanie Decroix introduced it, the gut is a fascinating organ, and it's a very complex organ. So this organ makes a barrier, like you can see here, between the external side of the body and the internal side of the body. This barrier is composed of an epithelial monolayer, composed of several cell types, mainly enterocytes, but you can find also here in blue goblet cells, secreting the protective mucus. And in the crypt, you have stem cells and proliferative cells that allow the tissue to regenerate. So as you can see also, this monolayer is organized in a 3D manner, alternating crypt and villi in the small intestine and crypt in the colon. Also, in addition, at the basal side, the immune cells, and at the apical side, the microbiota, altogether also ensure the good function of the gut. In addition, this organ is permanently exposed to two mechanical forces. One induced by the fluid flow, inducing a shearing force, and the other one induced by the peristaltic motion, inducing a cyclic stretching to the tissue. And these two forces are important for the tissue function. However, in a context of host-pathogen interaction, the role of this force has not been well addressed, especially due to the fact that we were lacking some important technology to modulate each of these mechanical forces. And it has been possible to test this more recently thanks to a new technology named organ-on-chip. So Stéphanie Decroix yesterday already introduced this technology, and it's very good because I do not have to introduce it again. However, here I will only talk about one specific organ-on-chip device that she named a dynamic organ-on-chip one. So this organ-on-chip is microfluidic chips that is made of a PDMS material that is a totally transparent material that is very practical to image it. And this device, this chips, is composed of two chambers, an apical and a basal chamber. At the apical side, you can grow epithelial cells, and at the basal side, you can also grow other cells, such as blood vessels, or you can even flush some immune cells. Importantly, in this device, you can apply all the mechanical forces as found in the gut, and you can modulate them as you wish. You can have a different flow at the apical and basal side, and you can modulate it each one. And thanks to these two lateral chamber, you can cyclically stretch the tissue to recapitulate the peristaltic motion. So with this device, you can do a lot of different things, such as imaging, but as well, you can also recover all the fluids that are flowing through these chips, and you can perform a lot of different tests. But today, I will, most of the time, show you example of using these chips by looking at fluorescent imaging or scanning electron microscopy. So with this kind of device, you can grow some different cell type, and one of the first things that we did was to use the classical colonic cell line CACO2 cells, that is his human colonic cell line, and after 5 to 6 days, the cells are fully polarized and organized, as you can see, in a 3D manner, alternating crypt or villi-like structure. Here is another example where we CRISPR-Cas CACO2 cells to obtain a CACO2 gene edited with ikedarin GFP. And you can see here the organization of these cells in these chips, where you have fully polarized cells that are organized in a 3D manner. Here what you can see is the top view of the chips, where we image it in live, and we applied stretching at a 10% stretched cyclically with a 0.15 Hz frequency. And here in this black area are seen the crypt. So what you see here is the 3D structure stretched. So with this model, we decided to investigate host pathogen interaction, and for that we started with a well-known pathogen, Shigella. So this bacterium is it's a gram-negative bacteria that is a human-restricted pathogen that is invading solely the colon tissue. It has been very well studied in the past, but always using 2D cellular model. In addition, because it is a human-restricted pathogen, it was very difficult to find an in vivo model to study it. So we were very happy to test the infection process using organ-on-chip system. So from the previous work in different labs, we have a model of its route of infection. So this bacterium can be ingested by contaminated food or water by the human body, and it reaches very quickly and specifically the colonic tissue. Then, facing the apical face of the colonic tissue, Shigella is supposed to cross this barrier through M cells. Once it has crossed this barrier, the bacteria could enter back to the epithelial cells, proliferate inside the epithelial cells, and cell-to-cell spreading, like you can see here. And finally, it multiplies and propagates all through this monolayer, and this leads to a massive inflammation that will finally destroy totally the tissue. So also, in addition, what was known from some studies in vivo, meaning in human patients, we know that only 100 bacteria is sufficient to promote the disease. And the disease is a quite important one. It causes dysentery that is still a very major health issue, especially for children. So we were still puzzled with this model. We were not totally satisfied because first we were missing the early stage during the infection, because here it's only a model, and we were puzzled by several questions. In fact, we never succeed to recapitulate the high infectivity of Shigella in even using human colonic cell line in 2D or not in 2D. And we were wondering why we were not able to reproduce this high infectivity of this bacterium. Also, another question that was also related is that we know that Shigella is a totally immobile bacteria. But it was a bit strange because Shigella is a very close relative to Escherichia coli that has this motility device. But during the evolution, Shigella gets rid of all flagella or motility device. In addition, Shigella is a non-adherent bacteria. So how this bacterium that is devoid of any motility device or any adhesion can really reach specifically and quickly the colonic tissue and invade it. So this was the question, and we were wondering whether the 3D organization of the gut and the mechanical forces encountered in the gut could help or could be involved in Shigella invasion. To test this, we compare the infection efficiency in the two devices. So either using CACO2 cells polarized in a 2D manner using a transwell system, well-known one, or we load CACO2 cells in these chips and fully mechanically stimulated it to organize the epithelial layer in a 3D manner. And then we apply different bacterial loading. Here it's written multiplicity of infection, either 10, 1, or lower. And we compare the infection efficiency in the different conditions. And first, in a transwell system, as we knew already, the infection efficiency is very low, with 1 to 2% when you put a high bacterial loading. But in contrast, when we do exactly the same infection, from the apical side of the chips, with 10 times less bacteria, you do see that there is almost 80% of infection. And we observed that to reach the same level of infection as found in a 2D monolayer, flat monolayer non-stimulated, we have to reduce the number of bacteria by 10,000 times, which was very impressive. So more or less, we could recapitulate the high infectivity of Shigella as found in human patients. So next, the question was, why Shigella is 10,000 times more infectious in a gut-on-chip than in a transwell? It can be due to different things. First, it can be linked to the 3D organization of epithelium, because in a transwell it's a flat 2D monolayer in contrast to the intestine chip. But it can be also linked to the mechanical forces that we applied. So either due to the fluid flow, the shearing force, or the stretching, cyclic stretching. Moreover, these two parameters, the 3D organization and the mechanical stimulation, could also, of course, could impact on the tissue organization, but as well, it could also trigger bacterial virulence, and this was not known. Indeed, we know that Shigella has many virulent proteins that are not activated in a constant way. It is activated in a timely manner. We know, for instance, that Shigella, I don't know if you see well here, but here is a tissue also with, and you see here in orange, red, the bacteria. We know that the bacteria can be first activating its virulence factor once it's adhering, let's say, setting on the plasma membrane of epithelial cells. But then after that, the virulent factor are switched off when the bacteria is entering inside the cell. And then, once it spread from one cell to another, it reactivated its virulent factor. And we don't know what is the cue that triggered the activation of this activation. So we decided to investigate the role of these parameters and using a bacterial strain, Shigella strain, modified that express GFP once the virulence gene is activated. And we decipher the three main stages during the infection, which is the colonization, so the first stage, then the invasion into the cell, and then the cell-to-cell spreading, meaning the propagation. And we decided to modulate each of the parameters, the 3D organization, the fluid flow, or the cyclic stretching. So first, let me explain you what we observe for the colonization adhesion. Okay? So for that, we apply Shigella to the apical face of the chips, and here we used the chip that is maturated in a 3D manner, fully mechanically stimulated with both shearing force and cyclic stretching. And here to look at colonization, we apply a very short time, quite short time of infection. Here it was a 30-minute time point. And the first thing that we first decided to observe was to perform scanning electron microscopy to observe the adhesion or the colonization of the bacteria to the chips. And here you can see it's a dividing bacteria. I don't know if you see well here. That is, and we observed that these bacteria were very often into the crypt side of these chips. Here what you can see in the center is a membrane ruffle that is a classical signature of Shigella that is in its way to enter inside the epithelial cell. Okay, so this first observation was rather qualitative, but we decided to quantitate a bit more. So for that, we wanted to know whether this particular location of Shigella into the crypt side of these chips, as you can see here. So here in red you have the nucleus, and in green are the bacteria. So we quantitate whether Shigella has a particular, this particular location, and we observed that 70% of the bacteria were enriched in the crypt-like structure of this Gutten chip. However, this particular location does not seem to be an active process mediated by the bacteria itself, since we apply beads, micrometer beads, the same way than Shigella and the same time, and we also observed that the beads are enriched in this crypt-like structure of the tissue. So it seems that the 3D organization mediate and the mechanical stimulation mediate the bacteria to crypt-like structure. Then we decided to modulate, as I already told you, the different cues. So we either grow the chips in a 3D manner or in a 2D manner, and then we modulated each of the force, the flow, the stretch, and we counted the number of bacteria after 1 hour of infection, which is quite a short time point still. And what we observe strikingly is that the first thing that matters for the colonization is the 3D organization. As you can see, in a 2D organization, there is a huge differences in the number of bacteria in contrast to a 3D way. But when you compare at this time point the importance of the different forces, the flow or the stretch, we didn't see at this time point big differences. But then we were wondering whether we were not missing some important information on the speed of the colonization, because here it was only one time point and we could have missed some speed effect that could be linked to each of these mechanical forces. So we decided more recently to perform a live experiment of the colonization, so the early time point, and thanks to our collaboration, we could also automatically detect the bacteria here in white using a software that is called IC that allows us to observe the colonization in live and to quantitate this. So we compare the two conditions, minus stretching or plus stretching, and you can see that upon stretching condition, you have much more, the colonization seems to be much faster. And we quantified this and observed that the speed of colonization under stretching was enhanced by 2.5-fold. But then it reached a plateau at around 30 minutes, 40 minutes. That's why perhaps after 1 hour of infection, we didn't see that much of differences with our previous experiment. So for the colonization, we observed so far that the 3D organization of the tissue matters a lot, and the stretching condition also enhances the speed of colonization. So now let's go to the invasion process. The invasion means that the bacteria is entering inside the epithelial cells. So it's a bit later during the infection process. So for that, what we performed was to, the same way, we either maturate our chips in a 3D manner or in a 2D manner. We modulate each of the mechanical forces, as you can see there. And this time, so we apply always the bacteria to the apical side of a chip. We perform a 2 hours of infection, and then we add some antibiotics to kill all extracellular bacteria. After that, we stop the reaction, and we could then recover. We could then recover the bacteria from the chips and plate it on classical LB plate to quantify the infection efficiency. And we could observe, so of course the 2D condition is rather low in terms of invasion process, but if you compare the different conditions in a 3D chips, as you can see here, you directly see that one condition is almost like the full mechanically stimulated condition, which is the cyclic stretching. So it seems that the cyclic stretching enhances a lot the invasion process in this organ and chip system. So this is one way that we can quantitate the infection efficiency, but there are, we could also quantify this by looking directly at a cross-section of these chips after this time of infection. So in the upper panel, you see the stretching condition, and in the lower panel, the non-stretching condition. In red, I don't know if you see well, but in red are the bacteria Shigella, and you can see, I hope, that there are much more bacteria, intracellular bacteria, upon stretching condition than without stretching. So we can also quantify these images like this. So it was very interesting, but rather descriptive. And now we wanted to address more how the stretching force could impact on bacterial invasion. And for that, we perform a tissue rheology characterization of a tissue. So for that, we collaborate also with Jean-Christophe Oliveau-Marin's lab. So they have built a software to decipher how the stretching force could deform the tissue, as you can see there. And you can see that the deformation is not uniform in all the chips. And this leads to a zone where you have higher tension. So we could build with this deformation characterization the stress map of the gut tissue under cyclic stretching, as you can see there. So in red are the, from yellow to red are the zone where you have a higher tension to the tissue. And we observed that Shigella seems to be enriched in this higher tension area. So here is a zoom of what we observe, where we could see the stretching tissue and the bacteria in purple here, where you can see that it is more enriched in this area. So with that, we also investigated how the stretching force could impact on bacterial virulence. So thanks to this modified version of Shigella that switched on GFP upon activation, we could quantitate the number of activated bacteria among time upon the different conditions. So in red are the conditions when the tissue is stretched, and in blue without stretched. And you can right away see that there is an enhancement of bacterial virulence upon stretching condition. Moreover, and this is quite obvious, but we also observe a higher rate of bacteria colocalizing with actin cytoskeleton. And we know that Shigella invasion strongly depends on actin recruitment to its site of entry. So altogether, the cyclic stretching induces local membrane tension to the tissue, a higher rate of virulent bacteria, a higher rate of bacterial colocalizing with actin, and therefore a higher bacterial uptake. So now let's go to the propagation. So propagation means once the bacteria has entered, it spread from one cell to another. So for that, to look at this stage, we have to wait for a longer time. We just incubate the bacteria, so always to the apical side of the chips. Then we let incubate it for 2 hours. Then we put antibiotic to avoid any extracellular bacteria to be ungrowing. And then to observe only the cell-to-cell spread that is not, the bacteria is not coming out of the cell at this time. So in the upper panel, you have a transmitted light images of the chips not affected. And in the lower panel, you see the same kind of chips, but this time affected with Shigella in green, as you can see there. And you can really see after 10 hours of infection that the bacteria is really totally invading the tissue, totally propagating, and the bacteria even go through the basal channel of the chips. We perform also a movie on that. As you can see here, we image simultaneously the upper channel of the chips and the lower channel of the chips. And we observed that the bacteria can cross this lower channel beginning of after, let's say, 6 to 7 hours, it starts to reach this lower panel. In addition, we also complexify a bit our system, and we load, so as usual, at the upper side, the epithelial cells, and in the lower channel, this time we load neutrophils that are labeled in red, and we infect it with the bacteria in green. And we observed that the strong recruitment of neutrophils to the area of affected area of the tissue, as you can see there. So then, for this stage, we quantitate like before. So the propagation stage in the different condition, either in the chips in 3D, in 2D, with either flow or stretch or both. And you can see this time that in the 3D organized chip, this time it seems that the fluid flow, the shearing force is important or enhance the propagation stage. However, we don't know yet how this shearing force is involved during the propagation. So on this part of Shigella infection, we observe that recapitulating the colonic tissue matters a lot and make a huge differences for Shigella infection, since we could, for the first time, recapitulate the high infectivity of this bacterium as found in human patients. We observed that the 3D organization, the mechanical stimulation, helps Shigella invasion, helps the whole stage of Shigella invasion. First, the colonization strongly depends on the 3D organization, where the bacteria can be hidden somehow in the crypt-like structure of the gut, and this will give sufficient time to the bacteria to interact with the tissue. And then, this stage also is strongly enhanced by the stretching force. However, as I already told you, during this stage, this is a passive process. The bacteria is there, but it's not an active process from the bacteria. Then after that, the invasion process strongly depends on the stretching force. And it depends on that for different points. First, because probably the tissue, thanks to the cyclic stretching, is more induce some local membrane tension that recruits, or at least trigger Shigella enrichment to this tension. And this seems to induce the virulence of the bacteria, and therefore its uptake. Then the last stage, the propagation, is further enhanced by the shear stress, but we do not know exactly how it does it. So finally, for this organ-on-chip system, this first experiment with this pathogen, we were very happy to use this system that is quite practical. And we could see that you can revisit some old dogma about host pathogen interaction, because now we change a bit the picture about Shigella entry route. So since when we have been using these chips to investigate other pathogen invasion. And for instance, we have some work that is with amoeba that has not at all, that is not at all the same kind of pathogen as Shigella, because this is a parasite that is much bigger than Shigella, and that has not the same kind of virulence device. And still, we observe with this pathogen that also the stretching force is very important to destroy and colonize the tissue. I will not talk about this pathogen today, but I decided to talk to you about another one that you may know, of course, is coronavirus, SARS-CoV-2. So we decided to investigate the gut side of SARS-CoV-2. Why? Because it could appear as a bit anecdotic, but in fact, among COVID patients, many have gastrointestinal symptoms, and we do not know why exactly, and it is very not very well investigated. However, we know that in the gut tissue, the cellular receptor for coronavirus, such as AC2, and some protease enabling its direct fusion, TMPR SS2, are highly expressed in the gut, even more than in the lung. So we wonder whether the gut could be a reservoir of the coronavirus. Also, another also argument to investigate the coronavirus in the gut is that we know that in the human body there's a strong gut-lung axis where an alteration of the lung could affect the gut and vice versa. So for all these reasons, we investigate the gut side of COVID. And the first thing that we used was to use these chips with, again, CACO2, and we compare the infection efficiency in CACO2 in Petri dishes in contrast to gut on chip. And once again, we observe an enhancement of SARS-CoV-2 invasion by almost fourfold. So this time, we did cross-section on immunofluorescence of the chips, and this here is an example of cross-section that we can observe. You can see in purple are the cells expressing SARS-CoV-2, and you can see, if you notice this, that in contrast to the Shigella infection, SARS-CoV-2 infected cells seems to be more restricted to the upper side of the gut, more the villi side, in contrast to the crypt side. Also, what we could observe, if you zoom in a bit, is that SARS-CoV-2 induces a massive reorganization of the tissue, and especially the adherence junction. That should be, like you can perhaps see here in green, very strictly labeling the adherence junction in a quite clear-cut way. But when you have infection, you see that the adherence junction are totally perturbed. Moreover, we perform scanning electron microscopy in these different chips. So here in the upper panel, it's the non-affected chips. You can see 3D organization of the tissue, and you can see in a closer view the beautiful microvilli that are very well aligned. And in affected tissue, here it's an infection for 3 days, you have the tissue, the picture is totally different. You see a lot of cells that are shedded from the chips, and you can see as well that microvilli seems to be totally disturbed. And here, the small things that you see here are the virus. Okay, so we were interested by this, but we said, okay, perhaps CACO2 cell is a good model, but it's quite not physiological. So we decided to go towards a more physiological cell type. And for that, we use human colon and ileum resection to generate colon and ileum organoid, as you can see there, and we can load this into this chip the same way that we load CACO2, and then we maturated the chip for 1 week, and we could observe 3D organization and polarization. As well, this is a picture of a part of the chip that is imaged in scanning electron microscopy, and here it's ileum on chip, where you see some villi here, villi-like, here, and you see beautiful microvilli, and here the advantage of this primary cell is that you have the whole panoply of cells as found in the ileum or in the colon, for instance, like goblet cells, enterocyte, and so on. With this primary colon and ileum on chip, we perform again an infection experiment, and we observe that the infection was a bit lower than in a CACO2 cell line, but still we have an efficient invasion of SARS-CoV-2, as you can see here in the green cells in ileum on chip system. And you can see again, so here it's two days of infection, and you can see also that once again, the infection with SARS-CoV-2 is much more occurring in the villi side of the gut. Here is the, here is the lower part of a chip, and here is the villi side. And here you can see also that we observe that there is a propagation of a virus from one cell to another all through this colonic barrier. And once again, what we could observe is that comparing chips that is grown in a 2D with no force with chips in 3D with, let's say here, the shearing force, fluid flow, we have a 10-fold increase in the invasion efficiency of SARS-CoV-2. And this time, the stretching force does not seem to be so important, but the shearing force seems to enhance a lot the SARS-CoV-2 invasion. Moreover, what we observe is that once again, like in CACO2 cells, here are the green cells are SARS-CoV-2 producing cells. So, and we observe again that it makes a huge perturbation to the adherence junction, where we do not see anymore, or almost not, the beta-catenin labeling. In addition, in contrast to Shigella, for instance, that is able to cross the chips from the apical to the basal side of the chips, for SARS-CoV-2, we observe the replication of the virus solo life from the apical side. Here is scanning electron microscopy that has been colored here, and the virus has been colored here in orange, and you can see a beautiful SARS-CoV-2 replication from the apical side of this colonic barrier. So altogether, in this part on SARS-CoV-2, we observed that SARS-CoV-2, I didn't show you all the data, but can infect and replicate efficiently in human colon and ileum surrogate. We observed as well that the virus uptake depends on AC2 and proteases. It can enter either by endocytosis or direct fusion in primary cell or colonic cells. In primary cells, we observed that all the cells that were infected were differentiated cells in contrast to proliferative ones. In addition, we observed that SARS-CoV-2 affects intercellular junction, architecture of the barrier, and microvilli. And last but not least, we observe once again that the 3D topology of the gut and the shearing force this time enhance a lot the viral infection. So today I talked more in detail about two examples of pathogen that didn't evolve the same way to the gut tissue. Or at least the infection process does not occur the same way. So on one side, Shigella, which is a bacteria, is invading mostly the crypt side of the gut, and the stretching force seems to be the force, the mechanical force, the most important to trigger its invasion and virulence. And on its side, SARS-CoV-2 invade mostly the upper side of the gut, the villi side of the gut, and the fluid flow, the shearing force, seems to be the most important one. So we have still many questions about this. For instance, we do not know how physical force could modify the tissue organization, whether it allows a higher expression of some cellular receptor or some membrane factors, and how a pathogen could evolve to this particular environment. And these are questions that we are still investigating. So I hope I convince you that using organ-on-chip system can make a huge difference when we investigated host-pathogen interaction. And with that, I would like to thank people that has been working on that. So mainly, first, Alexandre Grassin that initiated the study, that was then followed by a student, Thomas Fogas, Valérie, that is always working with me, Julian Nigro for organoid, Aleš and Elisabeth for image analysis, and Eloise for biomaterial microfluidic facility, and we always like to thank Philippe Sansonetti. So before stopping, I would like also to advertise quickly that thanks to the SBCF, we are organizing with Stephanie Misrelanke, that is in the audience, a meeting, the second meeting of cell biology of coronavirus, that will be a hybrid meeting, so virtual or in person. And you can see the person that we invited, so please don't hesitate to register. It's in January the 27th. Thank you for your attention.
Thank you. Any questions?
Thank you for the beautiful talk. You are showing that when you perform cyclic stretching, so in the first part of your talk on Shigella, so it increases the rate of infection. So do you think this is kind of a passive process as well, meaning that when you stretch the tissue, you expose more crypts, or you extend the length of the crypt, and this is why you uptake more bacteria? So would you have the same with beads, for example?
Okay, so the beads will not go inside the cells, for sure. So it's an active process. For me, this stage, the invasion stage, should be an active process because the beads will not enter inside the epithelial cells. And as I told you also, the stretching induces a higher virulence of the genes of the bacteria. So it's an active process at this time, meaning that the bacteria does not modify the stretching condition, but the stretching condition induces. So the thing that we don't know yet is how it works exactly. We know that the stretching will induce a higher tension, and perhaps this will reorganize let's say, membrane composition, such as cholesterol, actin, or things like this. And this, we know, it is very important for Shigella uptake. And this might trigger a contact that is different for the bacteria that will trigger its activation. So I think it's an active process.
So thank you for your seminar, Nathalie. I have a question. So in a way, the mechanical environment of the gut is favoring Shigella infection. So is it something specific to bacteria that do not have adhesion and motility? Could you do co-infection with another type of bacteria that has these particular features and see if one is helped by these properties?
Okay, so this is why now we are investigating the pathogen to see whether each one has a different, let's say, evolution to this particular environment. And with the amoeba, which is not a bacteria, this amoeba has a stronger motility device because the amoeba is really the queen of migration. And it is also very strongly enhanced by stretching force. So the example with amoeba will say, yes, it behaves not exactly the same way, but also stretching force seems to be very important for the uptake of the destruction of the tissue. But for other bacteria, so we have been doing that, but it's very preliminary, with some other bacteria that could be pathogen, that has adhesion like E. coli, pathogenic E. coli, and it seems to not behave the same way. But I cannot really tell you because it's very too preliminary.
And if I can add just another one, and that's it. Just out of curiosity, could you grow different microbiota on your chip and see if they have a protective effect against infection with your pathogen?
Exactly. This is also what we want to do. So this is now we are also in our way. So I mean, there are so many things that we want to do. We cannot do everything, of course. But yes, this is exactly where we want to go, just to investigate a more complex interaction with microbiota, pathogen, and try to see the competition in between the different things. And we are, for the moment, before doing microbiota, because it's a bit challenging. First, it's quite complex to recapitulate microbiota, for sure. But also, we want to avoid an overgrowth into the chips. So for the moment, what we are doing is it's not a competition like this with microbiota, but we are using some microbial products that are supposed to be important to reduce the inflammation of the, let's say, infection. And for SARS-CoV-2, our preliminary data show that short-chain fatty acid, for instance, that is the good metabolite induced by microbiota, seems to reduce SARS-CoV-2 invasion. So yes, it could be very good to go towards this direction.
So thanks again for your great talk. It's just one question connected to the first question that was asked. So you show that by stretching, you facilitate the invasion of the bacteria. So is it the stretching specifically, or is it mechanical perturbation? In other words, for instance, if you compress instead of stretched tissue, will you favor the invasion?
Okay, so it's difficult to answer totally to the question because the device is not made to compress. So I cannot really tell you that. I haven't done the experiment per se. We can only do stretching, but compression and stretching is not that much different, I will say. So for me, I will say that it should not change, at least in this kind of recapitulated barrier.
Thank you very much. So maybe I can also follow up on the stretching part. So do you see, while stretching, do you see the increase in like the ruffle formation, which is kind of a sign of infection, or did you try to look on that?
So yeah, so this we would like to investigate this, but you saw that the ruffle condition, we can see that in fluorescence microscopy, but it's quite difficult to do that in live cells, especially with a good resolution. And that's why also I came here, because we are really looking for good objectives to have high resolution with very long distance. And this is not very easy to obtain that. But I cannot really say that. The thing is, Shigella is also triggering this membrane ruffle. So with Shigella, since the stretching help a lot, let's say, the virulence of the bacteria, we have much more membrane ruffle. But we haven't done the experiment, we haven't looked or seen that much of difference without Shigella. Without any bacteria, you mean? No, I'm not. Yes, for sure, but for sure there will be more, because there is much more bacteria that could be. Yeah, without stretching, there are much less membrane ruffle, for sure. But, you know, the thing is, the bacteria just is sitting to the membrane, and right away it is induced this. So as soon as you have more bacteria populating the crypt, then you have much more ruffle. So it's very difficult to uncouple the different things.
Yeah, I have, thanks for the talk. Here in the back,
yeah,
in the dark side of the room. Two short questions. I mean, can you imagine of complexifying the system by adding also panel cells? In this case, you'll have an extra layer of mucus, and this is shown to prevent a bacterial infection, right?
Yeah, so normally with the primary cells that we use now, we have panel cells. So yes, but then after that, for the mucus, this is a good question. This is quite tricky to image it because it's very fragile and constantly renewed. But we are now in our way to try to investigate the best condition to have a really good thick layer of mucus, and this is quite challenging yet to image it. But yes, we know that we have mucus, but to image it is quite difficult so far.
And to bother you again on this link between stretching and infection, can you imagine that maybe this stretching is disturbing or breaking the gap junctions between cells, and this will create an entry?
No, I mean, yes and no. I think probably it does not destroy for sure the gap junction, because we have done in the chips without any bacteria. There is no leakage of the barrier at all. We put a lot of different kind of like dextran, different side of dextran, and there is no leaking. So no, I will say no. And it's normal because the gut is encountering also this kind of stretching, and it has to not be leaky. So.
Yeah, thanks for your talk. I just have a brief question related to the neutrophils, because you mentioned that you try to recapitulate, right?
Mm-hmm.
So what about the dynamic of the neutrophil? Are they attracted on the bacterial foci or on dying cells maybe? Can you...
So we did only one observation like this, so I cannot really tell you more about how it is attracted. But yes, you saw that in the movie, neutrophil seems to be attracted to the point where Shigella was invading the tissue, in the Shigella foci area. Okay, but then after that, we don't know exactly what is the trigger that mediates this. But in human patients, we know that upon Shigella infection, there's a massive neutrophil recruitment. And this also leads to the inflammation of the tissue. Yeah.
Thank you very much. I was wondering about the microbead control, and if you tried with different microbead sizes and densities, for example, and if you noticed these differences.
So we did, because at first we had only very small beads, so we did different size, but it didn't change, different size. We didn't do a lot of different size, but we have, so we go from 500 nanometer to 2 micrometer beads, and it didn't change. Okay. But you may expect that a smaller one will not have exactly the same way.
And different beads causing or densities? And could we, for example, relate that to interesting properties to have for drugs, for example, to select a preferred location to go to, just with physical properties?
Excuse me?
With other properties, with densities, for example, or coating of the microbeads. Did you try, and could we relate that to specific physical properties of the drug that we could use to treat, to select an area on the gut, for example?
Yeah, I mean, I think it's feasible, but we haven't done that. Yes, of course, yes, we can do that. I mean, we can do it. It's possible to do it.
Okay, let's thank Natalie again. So our next speaker is Matteo Rossi. He's a group leader in Rose Institute of Biology in Nice. His group is focusing on morphogenesis and mechanics of epithelial tissues. So Matteo did his PhD in Marseille, where he studied tissue morphogenesis driven by mousing tool contractility and how it polarizes tissue. And then he moved to EMBL in Heidelberg, where he studied tissue mechanics during Drosophila gastration before he came back to Nice to start his own group.
Thanks a lot.
Thank you.
So first of all, I would like to thank the organizer for giving me the opportunity to show you what we're doing in the lab. And I really like MIFOBIO because I really think it's a conference that allows to join the community doing technology development and eventually the community, the biology community. And I think that the interaction of the two is very important. I think that great advances in science are always driven by great ideas, but that is the technology development that allows to eventually test these ideas. And thanks to technology, technology does provide fruitful grounds from which new ideas can emerge. So the title of my presentation today is Mechanism and Mechanics Driving Composite Morphogenesis. So life starts from a fertilized egg, which is initially a quite simple and round shape. Then the egg divides, cells proliferate, and tissue change their shape in order to give form to the mature animal. While life can take a shape in very many different ways, the cell mechanism that drive tissue morphogenesis are quite conserved in the animal kingdom, meaning that even by studying the shaping of a simple fruit fly, this provides a great opportunity to uncover and unravel the fundamental mechanism driving morphogenesis. So there are eventually 7 main fundamental processes that allow to transform the tissue in order to give shape to the mature animal. I call them transformation function, if you want. And this is tissue growth. Tissue can shrink, fold, thin, thicken, converge, extend, and twist. Scientists and communities around the world have been studying these different processes, and they figure out the very highly specialized mechanism allows this transformation to occur. So this transformation process can happen sequentially, one after the other, during the development of the embryo. But this is not always the case. it can happen, and often happen in the developing embryo, that multiple transformations happen at the same time. So what we're looking here is the dorsal side of a xenopus embryo, which is developing. And what you can see is that the dorsal tissue, which will form the neural tube, is extending and at the same time folding. So this raises really a very important question, which is how is it possible that a tissue can undergo multiple shape transformation when each transformation is driven by a highly specialized mechanism? So in other words, how is composite morphogenesis driven and controlled? In the lab, we indeed studied simultaneous tissue folding and extension, and this process is very important during gastrulation. Gastrulation is a process by which cells are translocated within the embryo so that they can reach the right place to form the right organ at the right time. To give you an idea, the process that separates the head and the S is part of gastrulation to make you understand how important this process is. There are other processes like neurulation that I showed you in the previous slides, and eventually tubulogenesis. You know that we are tubes, living tubes that allow to exchange energy from the outside, and these are essential, in fact, for multicellular life. There are also many pathologies that arise from misfolding and misextension. There are spinal cord, angiogenesis, kidney, and respiratory pathologies. And that's why for us it was quite exciting to be able to tackle this composite morphogenetic process. So in the lab we're studying two types of tissue folding and extension. There is the first one, we call it orthogonal folding and extension, or also tissue wrapping, by which, in fact, the tissue extends along a direction which is orthogonal to the screen, and eventually folds along the vertical direction, like a burrito that wraps around. And then there is another way of making, in fact, a tube and to extend and fold, is indeed the so-called tissue in pocketing, by forming a pocket, where the tissue folds and extends along the same direction, and that's why we call it uniaxial folding and extension. So folding and extension will both happen along the anterior-posterior axis. To tackle these two, we use two different model systems. We use the early developing Drosophila embryo in the case of the orthogonal folding and extension. So an extension happens along this axis, which is the anterior-posterior axis of the embryo, while folding happens along the orthogonal axis, which is the ventral dorsal axis. In the case of the sea urchin Paracentrotus lividus, instead, both folding and extension happen along the same axis, which is the vegetal animal axis of the sea urchin. So the first project has been developed by a PhD student in my lab, and now has also defended his PhD, is Alfie, and the sea urchin project is developed by a postdoc in the lab, Bart. So today I will mainly focus on the Drosophila project, but probably I will provide a couple of slides, or one slide, to give you a flavor of the Paracentrotus lividus project. So the Drosophila embryo looks like a rugby ball. It's like an ovoid embryo and has the anterior part, and often or always I will present you anterior on the left, posterior on the right, dorsal on the top, ventral on the bottom. I will talk about cross sections. So the cross section are the plane that divide the embryo like if it was like a ham. And eventually you will have sagittal sections, which is the plane that divides the left side from the right side, which is probably the plane you will probably never cut a ham or a sausage. So if we now look along a cross section, what we see is that indeed the Drosophila embryo, after 3 hours of egg laying, it starts to form a fold along the cross section. But when we look at the sagittal section, which divides the left from the right, you can see that after a small constriction, well, the ventral tissue starts to extend. And this is shown when the arrow becomes green. As you can see, that extends. So these two processes happen exactly at the same time. So how is this possible? So in order to untackle this, I will try to divide the problem in two parts. I will try to talk about tissue folding in the first part of my presentation, and then tissue extension in the second part. But eventually we'll see that these two things synergize one with the other. So let's talk about the first part, which is which mechanisms are responsible for mesoderm folding. So mesoderm folding is dictated by dorsal-ventral patterning genes, which are expressed thus along the dorsal-ventral axis of the embryo. And as been shown already in the 1990s, there is very important transcription factor which accumulate in the nuclei of the ventral cells. And thanks to this transcription factor, one is twist, and eventually there's a second one, which is snail. There's a process called apical constriction, by which, in fact, the apical side of these cells reduce in surface area. This network, this actomizing network that constricts the cells, it forms eventually a supercellular actomizing network. So the question is, are apical actomizing contraction necessary for mesoderm folding? So in order to test this in the lab, we decided to use a technique called multiphoton plasma ablation in order to probe specifically actomizing contractility. In the past, during my PhD, I've been working on this technique, and I could show, in fact, that eventually on cells that have at the junction actomizing networks and adhesion spots, well, if we shine a high-pulsed femtosecond laser along one of these junctions, we can now deplete the junction from actomizing, and this will eventually lead to junction relaxation. Another test that I've done using this technique is to eventually test the integrity of the cell, because one question would be, is the cell then killed, or are the membrane preserved? In order to test, in order to give an answer to this question, we injected inside of an embryo a caged fluorophore, which is a fluorophore that does not shine. But after UV light transmission, well, this cage fluorophore become fluorescent. So we can now, using again the multiphoton laser by using multiphoton excitation, excite the fluorophore in the cell and see if there are leakages from one cell to the other cell. And what you can see here is after exciting and uncaging the fluorophore, you see that there are no leakages in between the cells, meaning that we can now selectively cut the actomizing network at the junction without affecting membrane permeability. You can also activate eventually the other cells, and same thing, you see there are no leakages. So now we have a great tool that allows to specifically ablate the actomizing network in order to eventually to test the mechanics and the function of the cytoskeletal networks. So this is just a movie showing what I just showed you. So we decided to use this technique now on the ventral side, where we have this actomizing network that drives apical constriction. And as you can see, after ablation, there's a strong recoil at the ventral side. So here on the top you have the membrane signal, and here the myosin and the merge. So after ablation, you can see, we can still see the membrane. Indeed, the membrane are preserved. And eventually, the cells, after recoiling, they eventually reform the network. So there's a phase of actomizing recoiling, and then eventually a phase in which the network is restored. So these cells are still functional up to a point that they can even restore the network, and eventually they can rescue the fold formation. So now this is the experiments we have performed. We try to perform with a on view fast the laser dissection, and then monitor along cross-section the curvature of the tissue in order to monitor the folding of the tissue. So this is the initial state, T0, where we can measure the curvature of the tissue, which is eventually convex. And after accumulation of apical myosin 2 that drives apical constriction, you see that the curvature of the tissue changes and becomes concave. In this graph, it becomes thus negative. At this point, we do laser dissection at the apical side, and you can see that the surface from concave, it becomes convex again, which is shown here with this time point tool, which is right here. Eventually, after actomizing a recovery, you see that the shape of the tissue again goes back to convex. So this experiment directly shows that the apical constriction and the actomizing network generates forces that allowed indeed to initiate the folding of the tissue. We then decided to try to perform a repeated dissection, since we know that this network would eventually reform, to see if sustained ablation would eventually completely impair the internalization of the ventral fold. And this is what exactly we did here with a pattern of beam, always using infrared femtosecond. We performed continuous and repeated laser dissection. So what you will see over time, so we did this for 15 minutes, and you will see over time that indeed the cells at one point will not longer be internalized. So this reinforced the idea that apical constriction, atomizing contraction are necessary to drive the folding of the tissue. So there are different models that have been explaining how this tissue can eventually fold. And one main model, which is now, I think, dominating right now, is the potential differential apical basal tension that would eventually drive a torque that would induce a bending moment. So it's the difference between eventually the tension at the basal side and the apical side of this tissue. Nevertheless, when we look at a specific mutant, which is a cellular mutant, so with Drosophila, one of the advantages, we have a vast number of genetic techniques, and one of these mutants that we have allows to have an embryo without any cells. So here you can see the basal side in the wild type case, the basal side of the cell on the apical side, and here there are no cells, in fact. So it's like a big syncytium. Well, in the case of the big syncytium, you still have indeed the changing curvature of the ventral tissue. So this shows that it cannot be apical basal tension difference, because here there's absolutely no basal side at all, right? So the model that I presented to you just a slide before cannot account, in fact, for this mutation and for the result that we get from this mutation. So the question here, then we said, what is the mechanism then that could eventually drive, in fact, the bending of this ventral tissue? So we then decided to model the embryo as one very thin elastic sheet, a bit like the mutant that we have here, right? That it has just the outer elastic network. And we decided to have a zone of pre-stress on the ventral side, which recapitulates the distribution of myosin 2. So this is distribution that goes along this direction here. So it's higher myosin 2 here and eventually less myosin 2 here. This is being measured also on the wild type. And when we minimize the energy, then we have large AP stress along the anterior-posterior axis because of the organization of the pre-stress zone, and eventually large DV strain along the dorsal ventral axis, which is along this axis. And by looking then at the shapes of cells and of tissue, we can see that we get more likely the same shapes changes that we get in the embryo. One interesting result that this model could provide is that what this model started to one of the results that gave is that the fold starts before in the middle of the embryo, and it's deeper eventually in the middle of the embryo compared to other zones, which are more posteriorly and anteriorly located. So that's why we went back to the real embryo, and we started to look at how these folds form along the anterior-posterior axis and not only in the middle of the embryo. So we not only monitored the central cross-section, but we started to monitor all cross-section from the anterior to the posterior axis. And we started to look at the folding along all this cross-section. So what you can see is the folds initially indeed starts in the middle point and then eventually propagates towards the posterior and the anterior. So you will see it start here and then goes here and then here and then here. And indeed, the folding will be deeper in the center of the embryo and less deep at the periphery. And this was never reported before. So we started to measure all these folds along all these cross sections. So what you will see here is that in blue are the cross section along on the anterior part, on green on the posterior, and lighter color are the one which are more medial, and darker color are the one which are more distal, which are more posterior and anterior. So this is furrow depth. So all curves start at 0 because I'm measuring the depth of these furrows with respect to the surface of the embryo. So that's why all these curves starts at 0. So this will be this point will move in, and this will be this point which will move in. So interestingly, you can see that the lighter colors are the one. So in the central zone, which corresponds to the lighter colors, is where the furrow will start invaginating before, while the darker colors, which are more the peripheries, is where the fold will start invaginate later on. And what you can see that eventually the ventral fold will be much deeper than the other folds. I then decided to plot this graph in another way by taking the same reference point. That could be, for instance, the plane on which this embryo is sitting. If now you plot the same graph in that way, what you can start to see is that there's some kind of a coordination that is happening along all these furrows. So this coordination can be graphically represented by a point, which is trying to follow, in fact, the central cephalic fold, which is then extended to a horizontal line. And what you can see is that the horizontal line is now following all the furrows at the same time, as if all these furrows has some kind of a coordination among them. So as you look, you can see that the line is going through the central furrow, but at the same time is also going through all the furrows at the same time. So at that point, we thought that maybe the right way of looking at this process was looking at the sagittal plane, which is the plane that I told you before is separating the left from the right, and eventually is going through all these furrowing points, which is exactly this cut here in the embryo, which is not a usual way in which normally this process is looked at. And when we look at this embryo in that way, what we can see is that what the tissue is doing over time is indeed flattening along the anterior-posterior axis, while on the cross-section is, of course, forming these folds. This is showing you live. So you see the ventral tissue is flattening, and then along cross-section you see the folding which is taking place. And when we went back to the model, so what we started to do here now is to measure changes in curvature not only along a cross-section but also along sagittal section. And interestingly, when we measure the changes in curvature over time in our wild type and in computational model, we'd had a quite good agreement between the two models. Interestingly, also, the acellular embryo generates a flattening of the tissue while it's generating this slight bent or folds on the cross sections. Another interesting prediction that model had is that the poles regions, in fact, they start to move inwards, and this color represents, in fact, the distance of the pole zones from the vitelline membrane. The vitelline membrane is like a cocoon which is surrounding the embryo, which is fixed and never moves. And what you can see is that the pole zones, they're moving inward so that the space between this cocoon and the tissue is increased. When we look at the real embryo also, we could see that the pole zone were moving inwards on both sides and creating larger space between this enveloping membrane and the tissue. The second other thing is that the model showed that there was much higher tension along the midline of the ventral fold, as you can see by this force, which is very high along the midline going along the anterior posterior axis. And by measuring tension along DV, that you can see here, we could also see that really at the midline zone we had the highest tension. So the model that we came up to together with the computational mathematician is that it could be that the polar tissue are indeed acting as a floating anchoring sites on which the ventral tissue is anchored to and constricts in order to drive the flattening. So we tried to think of an experiment in order to test this anchoring idea. And one ideal way would have been, in fact, to eventually generate ectopic anchoring points. In order to do that, we used a multi-view light sheet microscope that allows, at the same time, to visualize the entire embryo, and eventually also to use a multiphoton laser that allows, in fact, to generate tissue cauterization that eventually can allow to stitch the cell to the vitelline membrane, which is this fixed envelope. In this way, we can really generate ectopic anchoring points over which cells are eventually anchored. So one ideal experiment would have been, for instance, to generate two ectopic anchoring points, for instance, into two zones, and what the prediction would be that eventually the flattening will now connect the new two anchoring points. And this is exactly what we get after generating cauterization and fixing the tissue in these two zones. Now you see that the flattening is no over parallel to the anterior posterior axis, but now follows the anchoring sites. So overall, the idea that we have is that there's a supracellular contractile force which emerges from this, from the surface of the ventral side of the embryo that works like a cheese cutter, which then eventually forms a furrow. So this furrow formation is just the initiation of this fold, because then eventually the tissue moves towards the interior. And when you look at this acellular embryo, while at the beginning you do see the formation of this furrow, you see that the fold does not happen, meaning that still you probably need the lateral and basal side in order really to move the tissue towards the inside. So in the past, in 2010, there was a work using a technique called video force microscopy that allows to infer forces based on myosin 2 distribution. And what they have inferred is that eventually also forces along the lateral side of these mesoderm cells may be necessary to drive the actual folding of the tissue. So we decided then to test eventually also these lateral sides. In order to do that, we again used laser dissection, dissected the lateral network, and measured the eventual recoil. And we could see that the lateral network also could recoil. We measured this recoil and measured the initial maximum velocity, and could see that before lateral myosin 2 recruitment on the lateral side, there was much less tension than when lateral myosin 2 was recruited on the lateral sides. Nevertheless, while it was quite efficient to use laser dissection in order to test the apical network, as I showed you in the previous slides, using laser dissection on the lateral side is quite complicated, because in this part of the cells, of course, you have a lot of other components like the nuclei, eventually the microtubule networks. And while laser surgery is quite spatially temporary specific, is not protein specific. And that's why we decided to use now optogenetics in order to specifically, in fact, affect myosin 2 on the lateral side. So optogenetics is a technique by which, by using light, we can now change the conformation of a protein that will eventually bind to a receptor protein, which is A here, bind to the cortex of the cells, to the cell membrane, and this photosensitive protein will have eventually a cargo, a protein of interest, then when brought close to the cortex will modulate, or upregulate or downregulate the activity which is at the cortex. So this is the pathway that goes from twist and snail, if you remember, is these transcription factors which are essential for the folding of the tissue, down to the apical constriction and myosin 2 activity. So as you can see, this pathway goes through the Rho pathway. So one experiment we wanted to perform is eventually to downregulate the Rho pathway by using optogenetics. So here what we have a Rho dominant negative optogenetic construct, and by using 2-photon activation, we could now downregulate myosin 2 on the lateral sides. And what you can see is that in the zone where we activated we activated the tissue, well, it's exactly in that zone where we can see that the tissue is not folding as fast as the other zone, the control zone, where there was no activation. So 2-photon activation allows to be very much spatial and temporal specific, while the optogenetic tool allows to be protein specific. We then decided to do another experiment, which is eventually to upregulate the Rho pathway by upregulating ROGF2 upstream of the Rho. And so here we use a ROGF2 optogenetic line, and by now activating the lateral tissue, what we could see is that we could recruit myosin 2 on the lateral side and at the same time bend the tissue. This can also be performed eventually on one single side. And what you can see is that after activation, this tissue starts to bend. So this shows again directly that also eventually lateral forces contribute, in fact, to the bending of the tissue. So finally, the answer to the question that we asked at the beginning, which mechanisms are responsible for mesoderm folding, so the conclusion we had is that apical and lateral contraction driven by actomyosin networks cooperate to fold the tissue. So we now go to the second part, which is the extension of the ventral fold, of the ventral tissue. So what we know already is that other parts of the tissue undergo just simple extension. And this is, for instance, the lateral tissue of the Drosophila, which is the perspective ectoderm. So what this tissue does, it extends its length by a factor of 2 under the control of this gene called Eve, which is expressed in the zebra stripe pattern. So this process of extension is possible thanks to a mechanism called cell intercalation. So what happens, so these are different cells that have been marked with different colors. So the first row is blue, second green and red. So after 30 minutes, what you see is that blue cells, which were not in contact with red cells, eventually come in contact. And the tissue, the piece of tissue, you can see it doubles in length. So cell intercalation is possible via a process of junction remodeling. So what you can see here is that the junction between the green cells shrinks so that 4 cells come together in a 4-way junction, and eventually a new junction is formed so that the red cells can come in contact with the blue cells. So what we have shown in the past is that eventually actomizing networks are enriched along junctions that shrink in order to trigger cell intercalation, in order to increase tension and drive junction remodeling. In this tissue that doubles its length, what has been shown is that myosin 2 is planar polarized, meaning that all junctions which are parallel to the dorsal-ventral axis are enriched with this motor protein, so that cell intercalation is polarized in a way that the tissue will converge along the dorsal-ventral axis and eventually extend along the anterior-posterior axis. So we now decided to use again multi-view light sheet in order to try to do some comparison between this tissue that extends and the mesoderm tissue, which is right here at the ventral side, that also finally extends, and see if they had similar properties. So to do that, we had this multi-view system. So right now we're using a system from movie stream from Luxendo, which is then coupled to our own dedicated network that allows to stream data at 1000 megabytes per second and then process this data on a big workstation. So we can now image the entire embryo. So here you can see one single time point from dorsal to ventral, lateral left from lateral right, and then from anterior to posterior of a living Drosophila embryo. So with this data, what you can do eventually, you can then project the data as you would project, for instance, the world map, having United States on the left, Africa in the center, and Russia on the right side. And here you will have the same thing. So here you have the ventral side here, which will fold, as you know, and then on the 2 lateral sides, you will have the perspective ectoderm that we know they extend. And eventually you have here the dorsal side, which is shown 2 times just to emphasize the symmetry of the system. We can then play this thing, and you can see that the tissue folds, and eventually you have this other 2 tissues. So you can now see both ventral and dorsal side at the same time. So with this data, you can eventually track the 6,000 cells if you want. But what we're particularly interested here was at the gene patterns, and this is exactly what we looked in these carpets. So what I explained you up to now is that this lateral tissue, they undergo cell intercalation, eventually extension. And this is thanks to this gene, which is expressed in these zebra stripes, which is called Eve. But interestingly, what we see is that this gene is also continuing and is also expressed in the ventral tissue, which we know it folds, but now we know also that it extends. So the question here was, is the mesoderm doing similar things as the ectoderm is doing? And if yes, how is it doing it? And is it under control of Eve eventually? So in other words, one of the question is, do mesoderm cells intercalate? So in order to understand this, the first thing they have to know is that cell intercalation, as I explained to you before, is the exchange of neighbors, but this is not the full definition of cell intercalation because cells are not in 2D, they're in 3D. So it means that cells has to intercalate and not only apically, but eventually also laterally and also basally. And this will induce a full intercalation of the cells. And that's why we started to monitor cells not only at the apical surface, but throughout their apical basal length. And that's why this cell is exactly the same cell, it's just that it's being monitored at different depths. We can now look at this cell over time, and you will see that this cell indeed undergoes cell intercalation. This is shown in this fixed image. The red arrows indicate the time point where the cells undergo this four-way transition, and before it starts to undergo 4-way transition at 10 micrometer from the apical surface, and it will resolve in this intercalation, and eventually this will propagate basally and also apically, having a full intercalation of the cell. We can look at this also in 3D. So this is two red cells that will eventually come in contact, and the two blue cells which are already in contact. And this represents the surface of contact between these two blue cells. This is rotated 90 degrees, so you can see the width of the surface of contact. So you can see at 10 microns, this shrinks down, and eventually a new contact is formed between the two red cells. This is also shown in this animation where you can see this is the surface of contact between the blue cells. This at one point will shrink down, and the new contact will form between the two red cells. So we now measured all these intercalation which were happening in the mesoderm, and we can see that they're planar cell polarized, just like in the ectoderm. So it's the vertical junction along the dorsal ventral axis that shrink, and eventually the one which are along the anterior posterior axis that extend. And we could also measure that the number of cell intercalation in the mesoderm during this time window, during folding, was even much higher than what we had in the ectoderm. When we measured the way this intercalation happened, in the mesoderm, intercalation start to happen at 10 microns, and then eventually they spread along the apical and basal side. So this is time and this is distance from the apical side. While in the ectoderm, the intercalation was initiated apically and was then resolved basally. So this was something that made this mesoderm intercalation different from the ectoderm type of intercalation. So the question here was, which mechanism drives cell intercalation in the mesoderm? So what we know already from the literature is that when you form a tube, you can eventually start to initiate a cell neighbor exchange, so cell intercalation. And what you can generate, in fact, are these shapes called scutoids, by which cells which are, for instance, at the basal side, the yellow and blue cells which are not in contact, eventually come in contact at the apical side of the tube, while the green and red cells that get separated. This is possible in tubes because they have higher curvature on along one axis, but no curvature on the other axis, meaning that the distance between the green and the red cell, the center of mass of the green and the red cell, will stay constant since there's no curvature, there's no change in curvature between this direction and this direction, while the distance between the yellow and the blue cell, in fact, will become much smaller at the apical side because of the curvature of the tube. So every time you have a tube, you tend to initiate the cell intercalation. So we thought maybe is the fact that we're generating a tube by apical constriction, this could trigger the formation of cell intercalation. So in order to test this, we just took a very naive tissue and we generated ourselves an ectopic fold and tried to measure then the number of cell intercalation. So to do that, we again used the photooptogenetics, and we could then trigger apical constriction and generate a fold formation. We then measured the number of cell intercalation, and surprisingly we could see that in this ectopic fold, in this ectopic tube, well, we could not induce cell intercalation, meaning that this is not the mechanism via which this cell intercalation in the mesoderm are triggered. And that's why we thought that maybe there was some cell-active mechanism which was triggering cell intercalation. So we then look at the mesoderm cells at much higher magnification by using confocal microscopy. And what we could see is, so this is the apical side where we have the apical recruitment of myosin 2 that drives apical constriction. And these are the nuclei, and the basal side will be up here. So what we could see is that what we have is we have two tiers of myosin 2 distribution. While the first tier generates apical constriction, we didn't know what was the function of the second tier, which was located roughly at 10 micrometer inside of these cells. If you look at with high resolved images, you can see that this second tier is formed by the coalescence of the actomyosin network that kind of forms some kind of a pulse. And interestingly, if you see, you have some kind of a coordination between the formation of this pulse on one side and on the other side of the cells, as if, in fact, the networks on two different sides of the cells were some kind of they were biochemically or mechanically coupled. So these pulses are quite different from the pulses that were described in the past that belongs to each single cell, since these pulses emerge from the coordination of the contraction between neighboring cells. So we then monitored again this second tier of myosin 2 that we discovered, which is deep inside of the tissue. And by using fast spin in this microscopy, we could show that at 10 microns, the myosin 2 that we see in spots along cross-section, well, it distributes in a planar cell polarized way. So all vertical junction, like in the ectoderm, are indeed enriched in myosin 2. If we now follow one cell at 10 micrometer inside, we can see that during the shortening of the cell that initiates cell intercalation, we do have an enrichment of myosin 2. And indeed, the cells that have high level of myosin 2 are the cells that do intercalate, while the other junction that do shrink because the cell is still reducing in surface because of apical constriction do not induce cell intercalation. We also inject ROCK kinase inhibitor that inhibits the Rho pathway and inhibits the enzyme ROCK and inhibits myosin 2 phosphorylation, and in this case we completely abolish cell intercalation. So myosin 2 is indeed necessary to drive cell intercalation. We could also try to induce cell intercalation by using optogenetics, and we could see that by activating myosin 2 we could shrink the junction, showing that cortical tension eventually is sufficient to initiate cell intercalation. So cell intercalation is based on two steps. There's the shrinking of the junction. Eventually there is also the extension of the junction. And in the past it has been shown that the extension is possible via the interaction of the cortical network together with the medial network that allows junction extension. The problem is that in the mesoderm we don't have no medial network because this process here is happening apically in the ectoderm, but it's happening inside of the cells in the mesoderm. So we thought that maybe tension along the anterior-posterior axis could be some kind of a supracellular mechanism that could eventually drive tissue extension. After dissecting the network and reducing the tension along the anterior-posterior axis, we could see that we could, in fact, we didn't affect the shrinking process, which is a cellular mechanism, but we did affect the extension process, meaning that while the shrinking process is a subcellular mechanism driven by this planar polarized distribution of myosin 2, the extension process is driven by the contractile network, the supracellular contractile network, which is spanning the ventral side of the tissue. So cell intercalation is possible when cells have contact and adhesion spots. If cells are not in contact, they cannot remodel their junctions. And up to now, what we know is that junctions were only established in the ventral side, only at the apical side. The problem here is cell intercalation is not happening apically, but is taking place on the lateral side of these cells. And here, no junction were ever described before. So we now decided to perform an experiment in order to test if these zones, which are these foci, which are enriched in myosin 2 that trigger cell intercalation, are also point of adhesion. So to test this, we used a microcavitation bubbles that allow to generate ectopic contractions. So we generated an ectopic contraction in the neighboring cells, and what you can see is that this contraction pulls specifically on this node, and this really directly demonstrates that this point is a point of contact between two cells. We thus looked at epithelial cadering with high resolution imaging, and we could see that indeed these lateral clusters of myosin 2 also correlated with distribution of ecadherin. Contraction of actomyosin network, what they can do, they can cluster ecadherin in order to form spot adherence junction. And this can also be generated ectopically, because when we activate with optogenetic myosin 2 contraction by using ROGF2 optogenetics, we can also induce spot adherence junction establishment. So this can be also generated ectopically. So finally, which mechanisms are responsible for mesoderm extension? Well, these are polarized cell intercalation, which are controlled by the synergy between the anterior, posterior, and dorsal-ventral gene patterning that drives mesoderm extension. So I didn't show you the gene patterning part, but thanks to Drosophila, what we could do, in fact, is to use mutant that lack eventually the snail gene or the Eve gene and see how the extension and the folding is affected. So this is the genetic part that I didn't have the time to show you today. So finally, there's a two-tier junctional mechanism, a two-floor junctional mechanism, one at the apical site to drive apical constriction, and one 10 micrometer inside of these cells to drive planar polarized cell intercalation that allows to have simultaneous folding and extension at the same time. So I'll just provide one very fast slide on the sea urchin embryo. So this is a project that has been developed by a postdoc in the lab, and I decided to start using sea urchin because it has a great advantage compared to the Drosophila embryo, is that the Drosophila embryo is within this vitelline membrane, so there's no possibility to access the tissue to use, for instance, atomic force microscopy, micropipetting, thus measuring actual forces and mechanical properties. While the sea urchin embryo is quite accessible, and that's why we can now use, for instance, microindentation, as you can see here, or micropipetting, and given this transparency, we can also do full 3D segmentation of this embryo and look at the shape of all these 1,000 cells over time. So I here want to thank you for your attention, and I also want to thank my team. So I showed you today the project of Alfie, that was a PhD student in my lab that did the Drosophila project, Barthélemy together with collaborators in Villefranche-sur-Mer, Jennifer Crochet and Guy Lemont, and also at INRIA, Grégoire Melandin. We are now developing the sea urchin project, and Jocelyne Etienne, Philippe Marmottin, and Catherine Quillet at the l'IFI in Grenoble that developed the model for the invagination. I still want to thank also the PRISM facility, because most of the images you saw were done with multi-view light sheet microscopy, but many also with a microscope like confocal and spinning discs that are always freely available from the PRISM facility. And also thank the Luxendo company, because together with Région PACA, together they have financed, in fact, the scholarship of Alfie. So I now want to thank you again for your attention, and I'm ready to take questions.
Thank you for this beautiful presentation. So I have a question regarding your first part and the optogenetic experiments where you locally activate myosin 2 and you see that you trigger the invagination. But what happened in, let's say, in a normal condition? So do you think you have the same similar local activation, or you have a homogeneous distribution of the myosin and some local friction that allows this invagination? I was not sure to understand exactly what trigger without the optogenetics is local invagination based on the myosin.
So there is one interesting aspect about this invagination, is that, as I said, the invagination happens before in the center of the embryo, in the middle zones, and then eventually propagates on both sides. When we look at myosin 2 distribution, for instance, we don't see any wave of myosin 2 going from center to periphery. When we also look at the cell shape changes, we don't see that there's more apical constriction in the center than at the periphery. So there's no like cellular phase wave which is traveling, right? So it's more likely an embryo scale mechanism where you now have, of course, subcellular networks that eventually are interconnected together to form a supracellular network. And that's why we can see similar things also when we look at mutant where there are no cells at all. So from that point of view, what we think is that is just this elastic. So the system that we have, the model that has been developed, it's just purely elastic. So there's all the viscous part that has been modeled is not being modeled because probably of simplicity, because it's much more simpler to just have a purely elastic. And still this model, since also the process is very fast. I mean, this process happens in 15 minutes. All this invagination of 600 cells. So we thought that at the end, this purely elastic model would have been sufficient. And indeed, most of the feature that the model predicts are the one that we see, and every time when we see things, we also can see it in this model. So I don't know if this kind of answers your question. Yeah.
Hello. So regarding the mesoderm extension, so you said like in fact the T1 transition is responsible for this elongation. So I was wondering, like, I imagine that when you have a T1 transition, you have a relaxation, local relaxation in the tension within the tissue. I was wondering if you looked at the way that this transition can be transmitted to other groups of cells on the side. Did you look at the coordination of T1 events along this axis of extension?
Okay, I didn't specifically look. So in general, when I look at the T1 transition, I see that they are quite homogeneously distribution. So I don't see distributed. I don't see zones where there is higher density of T1 transitions. So the T1 transition, I think it's very sudden. So you have a T1 transition. You have a junction that appears. Because there's no junction eventually at this depth inside of the cell. So the junction is generated on the fly in order to create contacts locally. Then the junction is shrunk, and then from that point on, the junction eventually will disappear. Okay. So these, if you want, these local tension are very subcellular and transient in order to allow the swapping. So I'm not sure if this would have then a global effect on the entire tissue. I think that it's just a way to have a local swapping in order to facilitate the exchange of neighbors. There's another tissue which is at the posterior side of the embryo that also undergoes a movement which is called posterior midgut movement. And it could be that there's a coordination between the intercalation of these cells and also the posterior midgut movement that allows the eventual extension of the entire tissue. So I didn't tell you the entire story if you want today, but I think it's the composition of multiple parts of the embryo that eventually allow the extension, not only the intercalation per se. So I think the intercalation is necessary, and we know because we have blocked intercalation. I didn't show you the mutant. We block cell intercalation. We block extension, and the tube also becomes much wider because by converging the tube becomes thinner. So intercalation is necessary, but probably is not sufficient per se to trap the extension.
You don't see any temporal correlation between the T1 levels on the wave? You try to get that?
So at the scale of the entire embryo, what we see is that we have a high burst of cell intercalation in the mesoderm at the ventral side. And during that time window where the mesoderm extends strongly, the lateral tissue has very few intercalation at that time point. And this could be by the fact that the posterior midgut eventually also moves. So while the lateral tissue feels more torsion, the ventral tissue, which is located on the ventral side, will feel much more extension because it's right in line with this pole tissue that moves upwards. So it could be that the ventral tissue is the one that really needs to extend much earlier than the lateral tissue, just because it's in line with the extending zone right away. While the lateral tissue would eventually have some torsion at the beginning and then extend only during the second phase of the extension, where the posterior midgut really moves a lot on the dorsal side of the embryo. So I think there is some kind of a distribution along the entire embryo that kind of makes sense.
So thank you for our seminar. I have two questions. The first one is your embryo is very asymmetric, but how asymmetric does it have to be to initiate the process?
So the embryo is very symmetric.
Asymmetric. It looks like a Rugby shape with like...
You talk about anterior-posterior or dorsal-ventral symmetry?
I mean, they are anterior-posterior, but also both dorsal-ventral in a way. I mean,
so I think the greatest asymmetry that we see is really the dorsal-ventral eventual asymmetry in terms of geometry. Yeah. While it has a quite nice lateral-to-left symmetry. The anterior-posterior symmetry is very little. I would say that anterior-posteriorly, doesn't have a...
But could you simulate, for example, more symmetrical embryos and see the effect? I mean, genetically, I think it would be too complicated, but in your simulation, to change the shape of your embryo, to make it more symmetrical and to see if you...
So this is one of the experiments that eventually we were planning to do. So we have mutants in which, in fact, these embryos are like round and smaller. They're like balls. And of course, that would be interesting to see how the shape, the size, and the geometry of the embryo would eventually modulate this propagation of this fold at the ventral side. Yeah. So these experiments are possible by using eventually some mutants that are available. Yeah.
Okay. And the second question I had is, for your second tier of myosin 2 localization at 10 micrometer, how is it set up? Because it's very stereotypical in your cells. So what is the information that puts it there?
So we know that the apical basal polarity is essential to establish the zonal adherence, right, at the apical side. But this is not the only way you can generate junctions. To say the truth, these cells, while starting to invaginate, they express snail. And in fact, they're starting to lose their apical basal polarity also because they will undergo epithelial to mesenchymal transition. But there are other ways to generate junctions. So if you have a quite homogeneous distribution of ikaderine, for instance, at the cortex, even though it's not forming spot adherence junction, if now you can generate contractions, this can eventually, they can bring together ikaderine protein that eventually can come in cis and trans interaction and form spot adherence junctions. So I think, as I said, I was saying before, these adhesion spots are kind of generated on the fly. You just contract the network, and eventually this, which tethers with the cadherin protein, can then, densify this protein in a way that it will form junction. It's a very transient and in time and in space event that really allows to generate junctions just at the right place at the right moment, just to pinch the cells so that they can intercalate, and eventually this junction will just then disappear, because at the second tier then you will not have junction in general. So they're really ad hoc junctions for this process.
Very interesting results. I had a naive question on your last slide, but
the thanks slide.
The previous one. No, you said that the sea urchin has the advantage that cells are accessible, but actually the sea urchin has a hyaline layer which might have mechanical importance, or is that
so in fact there is not only the hyaline layer, there is also an extracellular matrix, so it's doubly covered, if you want, with some extracellular structures, but that's part of the system, right? It also eventually have cilia, that's part of the system. While in Drosophila we cannot even reach the tissue, right? Because it's encapsulated. So of course, this is the structure of the sea urchin. I agree with you. There is the extracellular matrix that eventually, and the hyaline layer, that both sustain eventually apically the cells, and maybe provide some mechanical properties which will eventually be essential for the folding. But it's part of the system, yes. And there are techniques to eventually remove the different substrates and eventually test the role of each substrate in the morphogenetic process that I'm interested in here, which is the formation of the archenteron. Yeah.
Actually, a follow-up question. So then, in the Drosophila, what is the contribution of the shell to the whole process of
So there is work from the Lequi lab that shows that sometimes these cells, even at early stages and apically, they can adhere and maybe walk or anchor to the vitelline membrane. For sure, what the vitelline membrane does is creating a zone where eventually factors can be secreted, and this factor can eventually diffuse. In many other systems, there is no shell, and you probably need maybe much higher densities of this factor in order to reach critical levels. So probably this could have a mechanical role, but also a biochemical role, right? It's difficult to test this because, as I said, for some reason there's high pressure in the embryo, and people have been trying to use different solutions to try to remove this vitelline membrane. I don't know if Eric managed to do this, but for now it's really impossible to do this mechanical experiment, in fact.
Matteo, so in the last part of your talk, you mentioned in these, when you were talking about these basolateral contractile units, you mentioned something about maybe the technique. You said you use microcavitation bubbles. Did you say that? Or
yes. So there, in the lab, we have mainly two techniques to generate this ectopic contraction. We have a raw optogenetic technique, and eventually we also can use femtosecond infrared laser. And when you increase the power, you can eventually generate small cavitation bubbles. And for some reason, when you generate these cavitation bubbles, after the bubble implodes, this locally creates some contraction. So there's some kind of a recruitment of actomycin that is created. So we still don't understand exactly what is the mechanism that allows to go from the cavitation bubble to the contraction, but we still use this technique in order to perturb neighboring cells and see eventually how these ectopic networks can pull on potential anchoring points. And here, as you could see, I mean, this network was specifically pulling on this myosin 2 focus.
All right.
So we still don't understand exactly how we go from the cavitation bubble to this ectopic contraction, but we see that it's quite efficient. So every time we have a cavitation bubble, often it's accompanied by a contraction of actomyosin network.
All right.
Thank you again. And now we have coffee break.
Thanks. Oui, il y a eu, je pense, 7 personnes. Ouais. Et ils étaient très contents. Ouais. En fait, on pensait d'en faire encore un cet après-midi, mais non, ça devient trop parce que je dois aussi, je dois partir parce que je dois faire tellement d'autres choses. Mais c'était très bien. J'ai fait toute une présentation sur laser tissue interaction. Et après, ils ont placé les embryons dans les capillaires, ils ont monté dans les movie spim, ils ont commencé à utiliser les movie spim pour faire de l'imagerie. Et on a fait de la dissection de laser avec ce système infrared. Il y a Spac Laser, cette compagnie bordelaise qui fait ces lasers super compacts. Ils ont l'EEM déjà intégré, ils ont un système chirp pour compenser l'impulsion. Et il est à l'intérieur du microscope. Au début, on avait quand même des lasers assez gros, maintenant ils arrivent à compacter assez bien ces lasers, qui restent quand même toujours très chers. Hein. Les prix. Hein? Oui, c'était très bien. Non, ça c'était très bien. So I'm not a genius of fixation, I would say. I would say I'm quite good with light imaging and fixation. I'm quite bad. I think I fixed a couple of times.
pas une clé USB. Non, je crois pas. Je crois que je l'ai.
Matteo, do you have the USB thing?
I didn't use it. I was using the keyboard, but it should be. It's on the back.
Que des choses comme ça. D'un côté ou d'un moment, ça va pas. Moi j'en ai marre de ça.
Ah, ok, I can try. Ok.
Peut-être politique, peut-être non, voilà, donc tu devrais avoir un peu.
Ah non, it's just mine. No, actually, I'm not sure. I didn't want to. Oh, you grew up? No, no. I grew up in Hungary. There are quite a few immigrants, and so from the name, actually, because there was this revolution in '56, and it's really like, I mean, now you talk about refugees, but at that time, like 100,000 people left Hungary and settled somewhere in Europe. So that's like a significant number. Yeah. Smart in physics, they are quite. So the training was good, and of course, when they emigrated, they found jobs. Let's talk.
Okay, great.
Okay.
So did you understand? Was it?
Yeah. Very good.
Was it easy to understand?
Yeah. Sure. Also, I'm not a specific development person.
So that's why I'm asking you, right? Because you're the perfect audience to know if it was understood.
So if you didn't explain the trick, how you observe the search.
I mean, yes, I know.
And actually, I was planning.
You're all experts here in CRG. Because one of the difficult thing with CRG is the fact that it swims. Also the fact, right? The problem with the CRG is it swims, right?
It has a sea.
It swims during gastrulation.
But I thought because I discussed with Alex and he said, yeah, actually the yelling here is a little bit annoying. I was wondering, more if you want to do AFM, or there is a risk what you measure is the yelling here, or at least that some of the yelling here might participate.
Well, it's like the system has other components, so you could say, okay, this is the apical site that I'm measuring, and then if you remove, you can say, okay, this is the cells, if you're removing both hiding layer and extracellular matrix. But it's just part of the system. I mean, this is how the system is.
But that's interesting. So this idea of confinement.
It has also basal extracellular matrix. There's apical extracellular matrix, there's hiding layer, eventually you have the cells. basal extracellular matrix.
In the cavity?
Yeah.
Yeah, in the cavity I think there's always. In the mouse, in fact,
well, not in the Drosophila.
No, because it's system. They all apicobasilic polarized embryos like the one which are haploblactic. I think in fact there's always extracellular matrix on the outside that is the cells in mouse that we talked about.
And the other thing, we tend not to use AFM. We use microindentation. So we have points of 5 microns, 10 microns, 20 microns, not points of hundreds of nanometers. So it's much more like tissue scale more than subcellular analysis of. So that's why we don't use the FM. We use, as you saw there. I mean, the point was gigantic, right? So it's very big. And then we use micropipetting. We're using a lot of micropipetting because
so
that's
keeping the limit.
Everything. With or without. But micropipetting is the easiest thing. I mean, that's like super trivial. Microindentation is a bit more challenging. And then it's full of other mechanisms.
Excusez, c'est ma faute. J'avais pas mes lunettes, j'ai pas tilté. J'ai une idée de chose. C'est mon ordi avant de présenter.
With the switch, you can cut the embryo into parts, 4 parts, subparts. So you can really tear it apart, but then analyze each part while this is completely impossible with Drosophila. So this is why CRG kind of complements in terms of techniques what we can provide. Drosophila, all these techniques that you saw. I mean, I didn't show too many mutations. They had to go through all the genetic study that we did, but you can see it's still use quite many things of the genetic for synthetic morphogenesis, morphology, development technologies of it. It's growing more and more. These are typically the tools that eventually we have in Drosophila and many in other systems. We're not so much yet. Un seul doctorant. Oui, je l'ai mis. I make him work lots. But I gave him like a super great. I gave him organize a committee with Eric Michaud, Philip Eisenberg, Keller. So I put him a lot of pressure. So at the end, it was like super tense.
Was also in a partnership with the industry, so I guess it was working there as well.
So there was a lot of exchange, mostly in developing the technology and for the user interface, to optimize the user interface, to eventually have direction to optimize part of the hardware. So the company always needs interaction that's always the point. Once you get the images, you have to really try to see how you can then get these images and analyze the images, and not just have a beautiful image to show, but then try to extract the information that you need to advance with your project, right? And I think I've been working in technology development at the beginning of my master and PhD, and then more in fundamental biology. And I think having had a foot a bit in the two domains really helped me a lot also, to then apply very cool technologies and on very interesting fundamental biology as well. So I think it was an advantage for me to sort of work with, like in Fresnel Institute, where we did a lot of technology development, and then eventually with fundamental biologists, maybe unaware of the technology and more just in their biology. While I really think that you need both, because often biologists have a crazy idea and they would like to test, but there's no way of doing anything because they don't have the tools, the knowledge, and sometimes maybe the tools are available, but they don't know about them, or they don't know how to apply, or they don't understand them. So it's always good for biologists to come back to the people that really develop the technology to exchange at that point. It's the work, I think it's really a cooperative work that allows to advance differently from theoretical mathematics, where you just need your brain and your ideas to go on. But in biology, most often is very strong technological improvements that allow so many discoveries in biology. So we're a bit downstream of the technological developments, not always upstream. I mean, we're upstream in the sense that maybe the people doing technology, they know what people are interested in. They are the one providing them the tools to then do then the experiment.
So how have you been working on this?
Well, it started by applying to an institute which was doing sectioning, optical sectioning.
Hello. You will be the chairman.
Yes.
I read the paper about what you're going to present for this. I wanted to do the things nicely. Like I couldn't do in the end for everybody. But I think people should share that. No, but like in the end, like to be honest, I didn't know you before. And I found your paper like super nice animation, super nice examples in the videos. Yeah, I also decided to present it because it's a microscopy meeting in the end. So it's a bit older project, but for microscopy, it was a nice challenge. Yeah, but that's the thing also, like you kind of really put together like both microscopists and biologists. Yeah. Some conferences like they are like really like either pure microscopies or pure biology. Yeah. And here like really combine both. But I think that was very lucky. I moved to Göttingen, and there I'm also in charge of the facility. Yes. And then we also got some new microscopes, and for this project it was really perfect. So it was the perfect test for. And it's sort of because the electron microscopy part we did at the EMBL, but then some things we could not put together. But then some live imaging and super resolution really helped to understand what's going on, because with the EM you don't have the dynamic. So I was very happy with it. It took like a lot of work though to... Yeah, it was also a long project. So now, hopefully, I mean also there was not much happening over the last year. It was really not so easy, but I guess for everyone. You are in Paris? No, sorry, no, in Grenoble. In Grenoble? Yes. Oh, yeah. I work with Laurent. No, like Marcel Ballon. Yeah, okay. We know the whole weather.
Teaching one another, right? Biologists are teaching what their interest is.
And what are you doing?
I'm doing microtissues.
Okay.
I study the mechanics of them, and I use optogenetics to make them contract.
Oh, that's super nice. Yeah, I think that's so. Well, I don't know. Sorry, yeah, I was just thinking because I mean we also worked on the cortex and so on, and
we didn't do optogenetics, but I think now people really figured it out and it's working so well. As Matin also showed, it's really contracting. Maybe we should.
Yeah, maybe of putting the moves for it together because it doesn't have like for the probes that already exist. Yeah, the probe is kind of you just need to find a way to put them in the cells.
Yeah.
But we are at the moment trying to implement new new probes into cells, and it's a bit of a struggle for the biologists to learn things.
Yeah, I know it's always a bit of a struggle. But it's super cool. I think this whole construction of the cortex, I think it starts to be, people start to understand it and it really gets very exciting.
Yeah, but with also too, that was we can play with it so much right now.
Yeah.
Like user with laser section.
Yeah, there was a talk yesterday or the day before. With the C elegance, it's completely denial and the active. This is
super nice.
That's like wow.
But at the same, like I talked to him afterwards, and he can't put optogenetics into smart biology, so he becomes very good at developing like exogenous sequences. So it's a real struggle for him, and kind of they know the words, they know the concept, because he can't it would be super nice to see like while here you do this, interact with the cells. Maybe only 10% but more time in the tissues. But general. I really like the way you
I think cortex is very nice.
So here, for instance, it's very, very simple. Trying to build up.
Also for imaging is good.
Yeah, but I'm sure that I could have done better in compared to.
So you do live or more fixed or both?
Do live myself, and since I'm at the like my tissue, you're always talking about those things. I can't really look at very nice structures. Very small. And also, like I need a really long working system. So that's kind of fun. But I can still do like very nice things.
Yeah. But in fixed, I'm really excited about this clearing and so on, so that you can do high resolution even if it's quite deep. But live, I still don't think.
No, like fixed, like we just bought like a very nice working training engine, like rapid clear, and it's improved a lot. Like the open magic.
But you use like oil objectives then?
Yes. Like I can, if I invert my samples, like I can get closer. Otherwise, like it's so thick, like it's a few millimeters. You can make some issues. But once fixed, I just press them.
Yeah, that's cool. Yeah. Okay, then we start.
It's supposed to. It's a moment.
When I talked to you last time, you were like, you were going to be in charge. Were you going to be in charge of
the facility?
All of the imaging
Yeah. So, I mean, I have to say the last 1.5 years was like, because I think we were all just ready to go. And I mean, for the facility, I think we did a great job, honestly. But
in all honesty.
In all honesty. But for research, it was just, you have the change of people, everything, and I was sort of ready to try to find people, and then they just didn't come, So it was a bit on hold. But I think now I'm full of optimism that now we can start again. And I'm still thinking what exactly to do.
because also, I mean, I'm sort of adapting to the environment, also because of the facility. We really sort of have to start with mammalian cells because, you know, that's what people anyway run on the system, so we have to prototype things, and then prototyping just for prototyping is very boring, so you rather have a project. And so, and I mean, we have very nice Microsoft. So, but I think I'm sticking to the nuclear envelope breakdown, and also look at this in mammalian cells and you know, how it's torn apart, how the membranes are rearranged.
You know the stuff that Matrikel was showing, it seems to be roughly like the same type of ARP2-3 driven process.
Yeah, I'm not sure. So no, I actually think it's maybe different, but I mean, but material science is also very interesting because, you know, there is this mechanosensing through the nucleus, and then, so okay, that's one. But then the other thing which is that he finds, you know, that if you crush the nucleus, then you induce DNA damage because you have this DNA activity in the cytoplasm. But for example, that you have to switch off in cell division because then it doesn't work. And all these kind of things, I think it's interesting to look at.
In Max's experiment, as far as I remember, you have the rocket at the front, and then you have this sort of ARP2-3 base pushing at the rear. It reminded me of what...
Yeah, I know, but I think that really seems like cytoplasmic.
Okay.
It really seems like cytoplasmic. So I think, I mean, actually doing lots of things, and I think our process, I think it might be really meiosis specific and oocyte specific. But and I think also you can attach also outside of the nuclear envelope, and that's also very important. But maybe that's the difference. That's maybe more this link complex, this mechano thing, you know, linking the nucleus to the cytoplasm. But yeah, I don't know.
Yeah, I guess that's a good, and I think having the high spatiotemporal resolution imaging, all of that would be ideal for looking at these kinds.
Yeah, and I mean, I think actually Buzz Baum likes that idea, and I also like it, is that I mean, obviously formin and ARP2-3 nucleated networks are very different in terms of density, but then basically ARP2-3 is so dense that like microtubules and stuff cannot go through, and I think that actually could be like a common theme, you know, like at different places. So if you already have actin, maybe, but then if you activate ARP2-3, you make it really a massive blob, which you cannot permeate. And I think that probably comes and I mean generally, you know, like this ARP2-3 is the ARP2-3 network really is different from a contractile formin nucleated network.
Is that true that you do get some microtubules that penetrate the lamina-podium, or do you think that they're on the top surface or the bottom surface?
Yeah, I think that's all, but like in neural growth forms, you really seem to have this separation.
Yeah.
But I think that's a really interesting topic to look more closely.
So one of the things I've been looking at is spindle reorientation, and there you have the astral microtubules. You know, people say they interact with cortical organisms, but not cortical, they're out the membrane. So the microtubules have to cross the cortex in some way.
No, and I think that
I find it a bit difficult.
And I think so cortex and, you know, all these networks are a bit messy because you have both ARP2-3 and formin, and I think maybe you regulate this and to look at more carefully into that, I think it's very interesting. Yeah, and I think with these things around the nucleus, it's also, you know, you activate ARP2-3, and then, I mean, it's a way to push also, but I think it also might have this role of really just create sort of a shell, which is in terms of.
Yeah, yeah. All right.
Go. Ok. Là, ok, ben, je vous remercie. Je vous remercie. Je sais que c'est le 11 novembre, que c'est un jour férié, donc c'est d'autant plus dur et que c'est la fin, c'est notre dernier jour. Pas tout à fait notre dernière session. Cet après-midi, il reste encore des tables rondes, des ateliers et puis ensuite une dernière session ce soir de séminaire. Et donc là, c'est la dernière plénière en revanche. La dernière session sera une session plutôt d'approche sociologique ce soir. Et là, c'est la dernière session, ce sont nos deux derniers intervenants sur les contenus, on va dire, scientifiques de l'école. Donc voilà, comme d'habitude. Et il y avait une petite annonce supplémentaire. Donc je vais demander à Loïc. Ouais.
Yes, quick one. Today at 2 p.m. in the Estrade room, the room that is inside of the restaurant, close to the entrance. You have to look a little bit. It's called Estrade. I'm demoing the IDEN software that we designed for the image denoising. So if you're interested to have a demo and maybe a tutorial, so you can bring your laptop, you can bring your data sets, and we can have a look at it all together. 2 p.m. until 3:45 p.m. today. Thank you.
And just a little reminder, I would like to ask you to remove your posters. Take them home or use them for another conference, whatever you want. But please remove them because we need to put back the metallic hangers. Thank you.
Thank you, Anna. Et après, une autre petite annonce, c'est donc vous avez vu, il y a différentes actions. Il y avait le Fab Lab, il y avait aussi dans l'Optic Lab donc des choses qui étaient portées par la communauté, qui est structurée dans deux groupes de travail, qui est prototypage et pilotage de périphériques et mesure et métrologie pour la microscopie, qui sont deux groupes de travail qui sont dans le RTMFM, et les collègues qui organisent ces groupes de travail et également donc plus largement les activités du RT, proposer de rencontrer ceux qui souhaiteraient éventuellement se rapprocher en particulier de ces deux axes. Il faut se rendre compte que ce qui peut être fait à MIFOBIO n'est fait que parce qu'on travaille entre les MIFOBIO. Donc il y a eu un gros travail fait dans ces axes, et ces axes permettent de construire du contenu pédagogique dont vous avez bénéficié. Donc si vous souhaitez davantage rencontrer les acteurs, et en particulier les ingénieurs qui sont dans ces groupes, donc ils vous proposent ils seront là de 14 à 16 heures, juste à l'espace Fab Lab qui est en sortie. Donc voilà, n'hésitez pas si vous voulez davantage connaître les contenus. Il y a d'autres groupes de travail du RTMFM, donc voilà, c'était le... Et ce soir, on va changer un petit peu on fera à 19h l'AG du GDR, puis la remise des prix, et puis après on aura le dîner et on fera le bilan ensuite, et on conclura par une soirée organisée par nos étudiants et postdoctorants, qui va se finir sans doute assez tard ou assez tôt demain matin. Voilà, je vous remercie, et merci, Marie-Émilie.
So now we will have the last two speakers of the mechanobiology session, Peter Lennart and Guillaume Charras. And also, I want to tell you that at 2 p.m. this afternoon, here in this room, we have a mini symposium, mechanobiology symposium, with Laurent Blanchoin and Hervé Turlier, and 7 young researchers in PhD, postdoc, and just had their CNRS concours. So please do come to listen to these young researchers, because they didn't have a lot of space, I think, during this meeting. So it will be a good opportunity for them to meet researchers and be able to challenge their ideas and their research. So at 2 p.m. in Salle, méditerranée, and now the two last speakers of this mechanobiology session.
Good morning, everybody. So it's my pleasure to introduce you Peter Lennart. Peter Lennart works at the Max Planck Institute in Göttingen in biophysical chemistry, and he works on oocytes of starfishes. He studies more precisely the meiosis of them, and he's gonna introduce us here like the nuclear membrane breakdown that is made by the actin, actually, compared to other systems where it's different. And for this, it would also be interesting actually for microscopists, since he used very nice techniques, and especially correlative imaging using both electron microscopy and super resolution microscopy. So, Peter Lennart, if you will.
Okay, hello, can you hear me? Yeah, can you hear me now? Okay, real audience, my God. I hope I still remember how to give talks. So first of all, thank you very much for the invitation. This is my first time at this meeting, and it's really an absolutely amazing meeting. It's made for me, because from the cell biology field, everybody's here, I know, and I wanted to meet for years now. And actually, a few years ago, my group moved to the Max Planck Institute, where I'm also in charge of an imaging facility, so I can also fully appreciate all the new technologies and workshops which are presented here. So that's really a paradise for someone who is in charge of a facility. Yeah, thank you very much for the invitation again. So, yes, as I mentioned, we are at the Max Planck Institute for Biophysical Chemistry. So, as a microscopist joke, we are two floors below Hell, so at Stefan Hell's department. So I think that's very nice for microscopy. And we are trying to really apply new microscope technologies to answer interesting biological questions. Here I would like to present a project which is a little bit old, but I think actually very nicely exemplifies this, using the microscopy technologies to answer some and visualize some interesting biology. So our topic generally is to look at cell division, but not in the standard form, which maybe most of you know better, like in a HeLa cell, in a process of mitosis dividing into 2 equal cells. Instead, we study cell division in a specialized form, which happens in oocytes, and it's essential to produce the fertilizable egg with the reduced genomic content. So this is called meiosis, and as you can see here, the oocyte is a very large cell, so this cell division is adapted to make basically the process work in this very large cell. It's also highly asymmetric, because actually once you accumulated so much nutrients in the oocyte, you want to keep all of it to save it all for the embryonic development. So this division is highly asymmetric, basically just extruding the extra copies of DNA in these unviable polar bodies and keeping everything for a single mature egg. So basically the question is how all the cytoskeletal structures, so we know actually that by and large it's the same mechanism, but somehow these cytoskeletal elements adapted to make this division work in this very large cell and to make it highly asymmetric. And that's what we try to understand. And one general understanding that came from actually over the years from our work is that actually actin the actin cytoskeleton really takes a much more major role in these large cells than we know from the smaller somatic cells. So in the somatic cells, basically, up to cytokinesis, it looks like that you don't really need actin, but in the oocyte, you do need actin already from the beginning of cell division. And we are using this somewhat exotic model system of the starfish oocytes because they are very good for imaging, but it turns out that several of the things we found may be in a bit different form, but actually it is conserved to other species. So actually the theme that by now it's very clear that this general theme that actin is important in meiosis in particular, I think that's very clear that it's true for mouse oocytes, in any other organism, zebrafish, whatever you prefer. So here you see the early stages of meiotic divisions in a starfish oocyte. In red you see the membranes labeled with the membrane dye, and in grays you see the different actin structures forming. And actually by now we understand most of these actin structures. We characterize these patches of actin which nucleated around chromosomes, and actually that's required to coordinate the capture of chromosomes later by microtubules. Obviously you see this network of actin filaments forming here in the middle of the nucleus, which is required for transporting the chromosomes. So actually, maybe he's here in the audience. So Philippon worked on how this, on the mechanism of how this network is actually contracting. But I will not present this paper because maybe it's not such a good paper. No. So Philip actually told me how to make bad jokes. So that was an attempt. You know, for German people it's difficult. Anyway, so I will talk about this actin network. So this actin shell, this bright structure which you see transiently just here, just at the time of nuclear envelope breakdown, which actually took a long time until we understood what it's doing and how it's working. So Masashi started on this, and Natalia continued, and now in Göttingen, Ivan, as a student, finished it. So as a little bit of introduction to the nuclear envelope, so the nuclear envelope is composed of a double layer of membrane, and there are nuclear pore complexes embedded in this double membrane which mediate the transport between the cytoplasm and nucleus. And this whole structure is stabilized by an intermediate filament network which is called the lamina. And when the nuclear envelope breaks, actually this process takes can be separated into two steps, which is a little bit confusing because people normally refer to nuclear envelope breakdown as the second step. But actually before that, there is a partial disassembly driven by phosphorylation of nuclear pore complexes, so the nuclear envelope starts to become leaky, but you don't really see that unless you specifically look for it. And this is then followed by a rapid rupture, which is normally what you see very obviously by microscopy, even by transmitted light. This is when the nuclear envelope is ruptured, and we don't exactly know how this happens, but it somehow involves forces. That's quite clear in different systems. In mammalian somatic cells, it's microtubules mostly that pull apart and rupture the nuclear envelope. I will show you that in the oocytes, actin again plays a major role in this. So just again to go in a little bit more detail. So actually during my PhD work a long time ago, I developed an assay to distinguish these two stages of nuclear envelope breakdown. You can just inject fluorescent dextrans into the cytoplasm, and you will see that the smaller dextrans can enter the nucleus earlier, which is an indicator for this earlier step, the first step of nuclear envelope disassembly. And then there are large dextrans which can only enter when the nuclear envelope is completely ruptured. So because we had this assay, actually, and we can quantify this, of course, and you see the different dynamics. And what was very clear that actin has something to do with the second phase. You see, the first phase is not affected if we interfere with the R2-3 nucleated actin, or if we inject a large amount of phalloidin to stabilize, so basically remove all soluble actin from the system. We get a very significant delay in this second phase, but the first phase is unaffected. So this rupture, this fragmentation of the nuclear envelope has something to do with actin. And then we did immunofluorescence, well, actually phalloidin staining, and we could visualize a very transient structure, which you have seen on the movie. Here I'm just showing it in the three-dimensional version, and actually you could see that it's on the inside of the nuclear envelope. So in green you see the nuclear envelope stained, but it's just underneath, right? And we also could visualize the R2-3 complex, which is co-localizing with this actin shell and actually later these patches around the chromosomes. So overall, and yeah, we could confirm that also with an R2-3 inhibitor, so we can specifically remove this bright shell of actin by inhibiting R2-3. So overall, we could confirm that this actin shell at the time of nuclear envelope breakdown is nucleated by the R2-3 complex, and it's somehow required for this rupture of the nuclear envelope. And then it's also important eventually to make meiosis work, so otherwise the oocytes don't form a mature egg. So we did some conventional electron microscopy, very conventional glutaraldehyde fixation, and that actually also very nicely visualized this rupture of the nuclear envelope, as you can see here. And there was a dramatic difference when we inhibited the R2-3 complex. Here you see the nuclear membranes remain largely intact if you don't form this actin shell. So we quantified this. You see that this fragmentation of the nuclear membrane is prevented by inhibiting R2-3. So that was Masashi's paper from 2014, that we could just show that this is somehow, this R2-3 nucleated actin shell is somehow required for the fragmentation of the nuclear membranes. And now I would like to show you how we went further and tried to address how this actually works. You know, so now we know that R2-3 is required for this, but how does it work? And actually, I'm not showing it in the order how we did the experiments, but that was actually a rather late experiment. But just to show you that if you do fixed samples by electron microscopy and so on, actually it's very difficult to put things in temporal order, especially if it's a fast process. So there, just recording one nice movie of the process can be really critical, because then everything sort of makes sense all of a sudden. So actually I'm showing it now first, but this movie we got during the revisions of the paper, and that was actually very nice, because basically the reviewer said, okay, now I believe it, you know, because that was exactly their concern, that how you know what is the order of events. So our model was that this rupture happens very quickly at the time of nuclear envelope breakdown. And the problem is that okay, there is a scale bar missing, very bad. But so this yolk vesicle is about 1 micrometer. So you can see that the spatial scale is quite small. So, but because we had our new facility setting up, we got a very nice ARI scan system. And actually that really just made the difference that we could resolve. This is quite deep, so this is like about 50 micrometer deep in the living cell, and also it's very fast. So this is 1 frame per second to scan. But actually now, and if you deconvolve it properly and everything, you can see initially these spiky structures appearing on the membrane, and then the membrane is rupturing, right? So you can really see that this is a very fast process happening right at this time, when actually this actin shell forms. So then we went on and did a few more immunofluorescence staining. So while the membrane is rupturing, actually the underlying lamine network is remaining largely intact, as you can see it here. So to go into detail, actually here you see the lamina largely intact, and then there is this actin shell, which is, it looks like it's nucleated in the lamina, and it protrudes filaments towards the cytoplasmic side. And now if you stain with the actin and the nuclear pore complexes, then you see that the nuclear pore complex containing membranes are somehow ruptured, and as if these spikes were going through. So you can see that here we are really hitting the resolution limit, so the structures are quite small. This is just standard confocal microscopy and deconvolution. So we could also play around with STAD a bit. I have to say the images look much nicer and convincing, but I think if one is honest, I think with the confocal microscopy, for the biology point of view, we already could answer the questions, but it's prettier with STAD. So you can nicely confirm that initially the lamine is fully overlapping with the nuclear pore complexes. And then while the lamine is remaining mostly intact, the nuclear pores somehow form these dense patches on top of it. And again, this is not happening if you inhibit the ARP23 dependent actin nucleation. So just to show a few more nice STAD images, you can really nicely resolve. Here you see actin in combination with this nuclear pore blobs. And I mean, this actually here, STAD really gave a bit of extra information. So I mean, at least for microscopists, this is nice. So if you remember, I showed you these bigger spikes by confocal microscopy, and by STAD actually we could resolve the individual filaments within those spikes. So what you see as one thing on the confocal actually is multiple actin filaments. So we learned something from STAD, but actually we still missed something, which is the membrane. So just technically we could not find a good way to stain the membranes themselves. So we always look at the nuclear pores, but not the actual nuclear membrane. So that was the real big challenge, and because of that we turned to electron microscopy. But before that, because it's a MIFOBIO meeting, so I thought I inserted here as a small detour that on our STAD microscope, actually not for this project but for other projects, we implemented a workflow. So it's a Nikon system with a nice camera on one side, just an Orca fusion camera, and on the other side we have this Steadicon system from Aberior. And basically we acquire images of large regions of cells, and then we transfer the image to ImageJ to do some processing, so in this case identify nuclei, and then we send back the coordinates to, we use NIS elements for this, to move the motorized stage to one of the coordinates, and then we send a trigger to the Steadicon to make a STAD acquisition, and then go to the next position and loop over and over again. So, and this was a nice project by student Dorian, and Milena did the biology part, the samples, and was supervised by Antonio, who is the staff scientist in charge of the facility. So just to show you, with this, overnight you can acquire a large number of cells. I mean, if you did it, you know, with STAD, the acquisition is relatively slow, so it's very boring to sit there. But in this way, you can just leave the microscope overnight and acquire cells. And if we zoom in, this is, so actually these are live cells, and so we didn't overdo it. So this is a relatively low stat power. So you don't see the details. So the resolution is not that high, so you don't see the details of the nuclear pores. But actually, we, as a proof of principle, we try to count the nuclear pores. And if you try to do that on a confocal image, it's really not trivial. But on this image, you can really count the pores automatically. So you, again, overnight, you get a very good statistics of nuclear pore density by doing this. And this seems to be okay for the cells. So this is a SNAP dye we use here. And we actually acquire like 4 Z-stacks, and the cells seem to tolerate that. Okay, back to the starfish oocytes. So as I mentioned, the problem was that we needed to see the membrane, and by light microscopy that was difficult. So actually that's actually the first part of the project if we look historically. So Natalia at EMBL working together with Yannick Schwab's electron microscopy facility, in particular with Pedro Machado, they developed a very nice correlative approach to find oocytes at this specific stage and look at them by electron microscopy. So as I mentioned to you, we had this assay for finding the time of nuclear envelope breakdown. So basically, by the ratio of the 2 dextrans, you can tell where you are in this process. And what is really nice, that you can look at these fluorescence from this next strand. What I show you here, these are so thin sections for electron microscopy, right? So you have to freeze quite a few oocytes and section them, and then you see where they are. And then you can pick the 1 or 2 which are exactly the right stage, which is that the small dextran is almost completely in, but the large one is still excluded. That's what we were looking for. So I mean, a little bit of detail. This is a high pressure freezing protocol combined with freeze substitution. Overall, and then we acquire these large montages, and you see all different strange membrane structures, which I'm going to go through now. Yeah, sorry. That's just to explain what you see here. So actually, we looked at immunofluorescence, and we see that the earliest actin structures appear like at the equator of the oocyte. So when we cut through, we see these 2 regions where there is something going on with the nuclear membrane. And we sort of see the temporal order spatially arranged in a single section, because it's sort of a progressive process. So what I would like to highlight is that the preservation is really beautiful. So high pressure freezing in this case worked very well. So you see the nuclear pore complexes. This is the intact parts of the nuclear envelope. You see in the oocyte there are tons of nuclear pores, because they also store the nuclear pores for the early division. But then, as you get closer to this rupture site, you see that there are some gaps in this array, and actually you see that where the nuclear pores removed, then this regular distance between the 2 membranes is also lost. So sort of the nuclear pores are clamping the 2 membranes together. And then as you get closer again to the rupture site, you see these spikes which are protruding towards the cytoplasm, and then you see nuclear pores at the base of these structures. And you see these blobs of structures which contain nuclear pores as well. What we were very delighted to see, that actually we can see actin filaments in these spikes. So the fixation quality was really quite good. And then now we could start to put this together with the high resolution immunofluorescence data. So, and actually now it makes sense. So what with immunofluorescence was really a problem that people usually assume, and we assumed as well, that okay, nuclear pores, we know that in oocytes there are plenty of nuclear pores, so we took nuclear pores as a proxy for the nuclear membrane. But you see that actually that's a very wrong assumption, because in this process what exactly happens is that you separate membranes which are free of nuclear pores, and you have then these clusters of nuclear pores at the base of the spikes free of nuclear pores, right? So and now it makes sense. So you see it's combined in different overlaps. So basically at the base you have the lamina from which these actin spikes are protruding, and at the base of the spikes you have these blobs of nuclear pore aggregates, right? But then the EM data shows that at this stage the nuclear envelope is still continuous, and these parts are free of nuclear pores. So we know that it's continuous also because actually all these like poppy seeds, these are ribosomes, and we see that before nuclear envelope breakdown they are exclusively in the cytoplasm. So you can see them here, but they are not yet in the nucleus. So I mean, the exact membrane reorganization is actually a bit difficult to imagine, and I'm not sure we fully understand, but somehow in this process you then sort of it gets escalated to the point that you push nuclear pore aggregates sort of inside, and you have the naked membrane still outside. And here in these regions the nuclear pores are very densely together. And I mean, this is very weird arrangement, but we see the same if we look by immunofluorescence. So it seems to be really like this. So what we don't see by electron microscopy, but we see by immunofluorescence that the lamina should be somewhere underneath here, and then you have these nuclear pore aggregates attached to the still intact lamina, whereas actin sort of pushing the membranes outside. And then this eventually leads to the destabilization of the nuclear envelope. Here again you see that you have a spike, you have these pore clusters forming, sort of moving inside. You can even see here how they might form. These are the pore-free regions. These are the nuclear pore clusters. They are going sort of inside, and then somehow the whole thing explodes. And basically what you have left are these pore clusters, and the naked membranes sort of disappear, and it seems like they incorporate into the ER. I mean, we cannot distinguish it anymore from the ER membranes. Okay, so again, this is consistent with that the end product is this structure where you see these dense clusters of the nuclear pores, which sort of form clusters. Maybe they are connected into a network. So these structures here. Okay. Yeah. So based on this is our current model of this process, where we think that here ARP23 nucleates actin on the nuclear lamina, which forms these protruding filaments, which somehow push on the nuclear membrane and basically separate the lamina and the nuclear membrane from another. And it looks like that it's maybe because the nuclear pores, I mean, this is known that the nuclear pores are attached to the lamina. So it looks like that the pores remain attached to the lamina, and because of that you sort of protrude naked membrane, pore-free membrane, and you end up with these nuclear pore clusters again attached to the lamina. So in the end you get a segregation of nuclear pore dense clusters and membranes which are just like ER. And it looks like that this structure is not stable, so it disintegrates at the end. Okay, so just to say that, okay, you probably wonder whether it's a starfish specific thing or it's a bit widely conserved. I mean, I cannot give you a definite answer, but at least recently, very similar looking structure transiently at nuclear envelope breakdown have been observed in Nematostella, which is a jellyfish. And it has been observed in other echinoderms, not only starfish, but sand dollars, which is kind of the flat sea urchins. And actually, there is an older paper where a similar structure seems to be forming in a polyhete worm. So across evolution, it seems like that it's popping up. So it might be something generally relevant. How much time I have? I have no idea. Yeah. Okay. I'm good in time. So, but I just wanted to finish by actually just sort of making a few points which I think really are conserved sort of building blocks in this mechanism. So this is also nice because both Anne-Cécile and Laurent is here in the audience. So what they did here is that they patterned, basically they made a pattern of an activator of the ARP23. And this is an in vitro reaction where you just activate ARP23 and add actin to it. And this is what you form. And I just would like to point out that this really looks quite similar to this. So basically, if we imagine that in the lamina, you activate ARP23, I would imagine that you should form a structure like this. So why this structure form is that basically ARP23 activates actin, but the filaments keep on growing where there is no ARP23 anymore. That's why you get this hairy structure, and that's what we sort of see it here. Of course, there is a mystery why they don't grow in the other side. This we don't know. So they somehow know to grow in only one direction. And of course, then this would be the next challenge for a PhD student, for example, find the activator of ARP23. So I think here we have a quite clear hypothesis. We are looking for an activator of an ARP23 that is bound to the lamina, basically, at this time. Then the second point is that I mentioned that what seems to be key here, that you separate spatially the nuclear lamina and the nuclear membranes. And this actually is known to be, so basically it's known that if you remove the lamina, then you destabilize the nuclear envelope. That's not a surprise. This has a main contribution to formation of micronuclei, nuclei also in mammalian cells. And I mean, it's still not clear why the nuclear envelope is destabilized if you remove the lamina, although I think you can, easily imagine that you have to keep the nuclear pores in place and so on to form a stable structure. I think mechanistically it's not understood, and what I think this mechanism adds to this, that actin can do this trick to separate the lamina and the nuclear membranes, and this maybe in other contexts also can serve to destabilize the nuclear envelope. And again, that's very important, but because if you destabilize the nuclear envelope, then you might get DNA damage as a consequence, and formation of micronuclei, and so on. Finally, going back a little bit to the oocyte specific part of the process. So basically, what I showed you, that during this process, what happens, that you segregate a membrane subpopulation which is full of nuclear pores from the ER, right? So you form these nuclear pore dense clusters, and you separate them from ER. And this might be actually important from oocytes, because we know that in embryos, you store extra nuclear pores in the form of so-called annulate lamellae. And basically this mechanism could be generating these annulate lamellae. So you generate these nuclear pore dense structures through this process by segregating the membranes. So yeah, I mean, just as a sort of personal note, it's actually an interesting story because when I started my PhD in Jan Ellenberg's lab, he was sort of in a vicious argument about nuclear pore disassembly. So basically, he showed that in mammalian cells, the nuclear pores just disassemble and disperse in the ER. Whereas the people working on xenopus oocytes, they could purify a membrane fraction which is specifically containing nuclear envelope proteins. So they said that there is some kind of vesicularization which separates these membrane populations. And I think we can now conclude that maybe everybody was right. So in the small somatic cells, for sure, the membrane, the nuclear pores completely disassemble because it's a tiny cell. But actually, our data exactly shows. So this to explain what I'm showing here. So this is a starfish oocyte. Sorry for this; it was an old EM picture with not so good fixation. But this is like half an hour after nuclear envelope breakdown. But you can still see these membrane structures full of nuclear pores, right? So we actually really see that in the oocyte, this membrane population containing nuclear pores is surviving for very long, and it's not mixing with the ER. So you can easily imagine that if you ultracentrifuge this sample, then you will be able to separate those membrane populations because they are visibly different membrane populations. So just to say that I think biology is diverse. Okay, so with this, I'm at the end. So I would like to acknowledge my former lab at EMBL, Natalia in particular. It's her project primarily, and the early part was done by Masashi, and Pedro was very essential for the electron microscopy part. And that's my current lab. Very recently grew with a few additional students. And Ivan was there from the beginning, and he helped finishing this project, basically. Yeah, and thank you for your attention. And if you have any questions, I'm happy to answer.
Does it work? Thank you. Beautiful talk. Is there a sense of whether the nucleoplasm is kind of pressurized before nuclear envelope breakdown?
Yeah. So I think that's something would be nice to work on. So what we see, it's quite clear that the cytoskeleton, actually microtubules also pull on in starfish, but I think they just not enough. You can really see. So if you depolymerize actin, there are still some forces left. But if you remove both actin and microtubules, you really see there are no forces left. But when the nuclear envelope breaks, there is a shrinkage of about 10% volume. So that means that there is colloid osmotic pressure or something like this in the nucleus, which is independent of cytoskeleton. But yeah, I mean, you can see it. It's like a balloon, and it deflates when it breaks. Yes. So there is a pressure.
Thank you for the talk. You said that the lamina was quite intact before the nuclear envelope breakdown. So I'm not sure about what I'm going to say, but I had the impression that the lamina was not homogeneous along its length, so you had some sort of variations in the thickness or something. So do you think this could be related to
I'm not sure. I have.
So on the first picture you showed, we had this feeling that it was not homogeneous. So do you think this could be related to the actin nucleation sites?
Yeah. So I'm not sure I have that slide. I have a quick look, but it's a perfect microscopist topic. So we think there is a structural change. So lamina is a meshwork. No, sorry, I don't have it. But lamin is a meshwork. And but it's a very dense meshwork. So with the nice TED images, if we deconvolve them, we can see that we think we see that the meshwork is becoming, the holes are getting bigger. but we really were not confident enough whether it's a deconvolution artifact or not, so we didn't show it. But it looks like that, so maybe we should go back and use a more fancier microscope or take another image. But it's really actually a nice example where pushing the resolution a little bit further probably would be able to answer your question.
So thank you for your talk, Peter. I have two questions. The first one is, do you have an idea of the force exerted by these spikes? Like, could you inject beads, for example, and pull on them with tweezers, or doing laser ablation after R2-3 inhibition?
Yeah, that's a very nice idea. So actually, some of you might know Timo Betz is in Paris. So he moved to Göttingen, and he's a master of optical tweezers. And we already injected beads in the nucleus of the oocytes last month. So I think we are ready to go.
Okay. So great. So the second question I had is that eventually, when you inhibit R2-3, the nucleus do rupture with a delay. First, how does it rupture, and is it really a problem for the cell if nuclear envelope breakdown is delayed?
Yeah. So, I mean, well, I think we sort of have an answer. I mean, it really, you can see that these membranes really remain intact for a long time. And basically, I mean, it's a little bit hard to really directly evidence this, but we are really quite confident that's what is happening, that the chromosomes get lost because microtubules get basically stuck in these large and intact membranes. So you know, there will be some ruptures happening probably by the phosphorylation-driven disassembly, and this is enough to equilibrate this pressure, so the nucleus will collapse. But you really see these large stretches of membranes, which you can see on the electron microscopy, and these really shrink but remain intact. So if you look 20 minutes later, it would be still these large membranes, and that's probably just prevents the microtubules to efficiently capture the chromosomes.
Thank you for a nice talk. My question is related to the force question asked earlier. I wonder, do you think there could be a pulling force on the side of these nuclear aggregates, and then acting spikes is a consequence of this pulling force because you need a balance, and then but the acting spikes acts as a segregation mechanism to segregate the nucleus to the side, and then you could proceed later.
Yeah, I mean, yeah, I don't know. It's always this question of who is pushing and who is pulling. I think, in a way, I would think it's sort of consistent with what ARP2-3 normally does, which is pushing membranes ahead. I think that's what ARP2-3 very often does, and I think this is basically fits into this. That you. Yeah. What drives that is a very good question. Yeah, that's right. So there is this membrane segregation going on, and how the membrane topology there looks like and so on. There, I think, I think this is where, again, there is a technical challenge which to see that in 3D, actually by electron microscopy would be possible, but we didn't manage because it's quite a lot of work, you know. Then you need to do serial sectioning and reconstruction. This we didn't manage in the end, or we are really missing a good marker for light microscopy for the membranes. We tried a few things, but nothing really worked. And then we could, by STAD or so on, we could resolve the 3D structure. That I would really love to see that, you know, how these membranes segregate into these pore-dense blobs and like ER-like membranes.
So I have on the first line, two fast questions. So the first one, we know that we have actin and ARP2-3 eventually also at the leading edge of crawling cells, and they don't break the membrane, while the nuclear membrane here is broken. So what do you think is the actual difference between these two types of membranes to make the nuclear membrane so sensitive to this actin protrusion?
The nuclear pores, I would say. So here I think you separate these membranes, and but the nuclear pores are holes in that membrane, and they are disassembling at the same time. So I think this can I mean, also topologically, I think that to me makes a lot of sense that if you, I think I even made a drawing for that. I mean, this is where I think again the 3D tomography or something like this would be, so one could imagine something like this, so you have, so I'm not saying that ARPU3 breaks the membrane. It's just that you separate it from the lamina, which is a stabilizer, and then at the same time the pores are disassembling. Not all the pores, but some of the pores are probably sort of popping open, and then you are, again, I don't know how to do this in 3D, but you can imagine that you can actually, topologically, you can convert it to a network, right? Because with these holes, you can just expand them and convert it into a network. So I think the key is that you, actually for this mechanism, it's also required that the nuclear pores are somehow falling apart. So and that's what creates sort of the membrane gaps. I'm not saying that ARPU3 like pierces the membrane.
Okay, so that my question is really related to what you just said right now. So if you now upregulate ARPU3 before the right time, did you ever been able to do that to see if then you cannot, in fact, if this spikes to not break the nuclear envelope?
I think that's...
so there is not sufficient, let's say.
Yeah. So I think that's certainly on the to-do list. So like if we catch up on this project again, that's... I mean, we would need to identify the activator of ARPU3 and I mean, and then we could do those experiments, and yeah, I think that's very interesting.
Okay, thank you very much, Peter. We can thank our speaker again.
Thank you.
So our next speaker is, and last for this session, is Guillaume Charras, who works in UCL in London, and is mostly interested in mechanobiology and the mechanics of cells, and more specifically, he's really interested in cytoskeleton and the actin cortex of the cellular envelope membrane. And today he's going to present us how to study the interplay between mechanics and cellular signaling using AFM and optogenetics.
The microphone. Well, first of all, I'd like to thank the organizers for the kind invitation. It's been lovely to be here. I do apologize for bringing the English weather with me yesterday, but thankfully that stopped. And I'd also ask all of you to turn your camera feeds off for the presentation. Anyway, my lab is generally interested in understanding the mechanics and shape of cells and tissues. And I'll talk to you about some work that we've done on use optogenetics to look at all of that today. So one of the most fascinating phenomena in biology is how cells and tissues change shape. So whether that be during cell division, when you create the mitotic furrow, or during gastrulation, when you create this deep invagination. And in both of those cases, what you need is a patterning of signaling initially that will give rise to a patterning of mechanical properties and stress. And this patterning of mechanical properties and stress together will give rise to a shape change. And later on, this shape change can then feed back onto all of these different phenomena so that you can iterate the shape change. So essentially what we need to understand morphogenesis is to understand how the patterning of signaling will give rise to a patterning of mechanical properties and stress, and then afterwards how that will give rise to shape change. And on this topic today, I'll first talk to you about some work where we looked at how signaling controls the cortical tension, and we've chosen to look at mitosis and the creation of the cleavage furrow, but that's something that would also be applicable to any change in shape of the cells, so for example in migration too. And then I'll talk to you about something a little bit related to that, which is how cortex mechanics controls spindle orientation. And this is something that's perhaps more applicable to obtaining oriented tissue growth in response to mechanical stress. So most of the intrinsic stresses that shape cells arise from the actin cytoskeleton, and in particular the actin cortex. And this actin cortex is just below the cell membrane, and it's a network of actin, myosin, and actin binding proteins. And it seems to be responsible for most of the mechanical properties of the cells, and in particular its surface tension. And as a consequence, it plays a fundamental role in anything that involves shape change. So, for example, you'll find a very nice actin cortex at the rear of these migrating neutrophils, or it plays a role during cell division and also more generally during tissue morphogenesis. So during mitosis, the shape of the cells is controlled by pattern signaling. So for example, in this anaphase cell, you've got a set of regulators of the cytoskeleton that is different at the pole compared to the equator. And at the equator in particular, we've got a number of different regulators of Rho GTPases, some of which increase Rho GTPase activity, and these are RhoGEFs, and others that decrease it. And all of them together will regulate the activity of Rho-A. And once Rho-A is active, what it will do is it will regulate the polymerization of actin and the activity of myosins, and that together will give rise to a change in mechanics. So essentially, here we're confronted with the exact same question that I started with, which is that we've got a change in signaling that's going to give rise to a change in mechanics, and that will then give rise to a shape change. So to try and tackle that question, we used optogenetics to control signaling, imaging to understand what's happening in terms of downstream effectors, and atomic force microscopy to look at cell mechanics. But the first thing that we wanted to ask is if the local differences in signaling that we observe, so for example the accumulation of RhoGEF at the cleavage furrow or MP-gap at the cleavage furrow that together regulate Rho-A, do these differences in signaling translate into mechanical differences? And for that, what we do is that we use atomic force microscopy, and we acquire measurements of the apparent elastic modulus along a line that goes from pole to pole at about a 0.5 micron interval. And our atomic force microscope is mounted on a confocal microscope so that we can actually image what's happening at the same time and try to do a little bit of correlative fluorescence imaging with atomic force microscopy. So in this image here, we've got a HeLa cell that's expressing myosin with an IRFP tag. You can see that you've got most of the myosin at the cortex. You've got some accumulation of myosin at the spindle poles, and that's something that no one seems to want to go and look into, but we consistently see it. You can see the nucleic acids just here, and this thing is the shadow of the AFM. So if I play this movie, what you can see is that the cell essentially divides quite happily, despite the fact that we're constantly poking it to measure the apparent elasticity of the cell. And the reason why I'm saying apparent elasticity, and I'm using this as a proxy, is that the models that we have for deconvolving the AFM measurements at the minute and extracting, let's say, cortical tension versus cortical elasticity versus cytoplasmic elasticity, they're just not there. We don't know how to do it, and so we just have to rely on the fact that we are sensitive to changes in tension, but we can't actually get you an absolute value. Okay, so with these data, what we can do is we can then try to visualize the changes in cortical myosin on chymographs. So in this case here, what we do is that we look at the myosin intensity along the contour of the cell, and we plot this on the y-axis, and over time we've got the evolution of fluorescence along this contour. And what you can see is that we start with myosin that's more or less homogeneous. At the time of anaphase, which is this red line here, we can see a very clear enrichment in myosin that takes place at the cleavage furrow, and that persists throughout cytokinesis. If we look at apparent elasticity, things are a little bit different. So it's initially very homogeneous. Again, we see this big spike that happens at the equator where the cleavage furrow will be just after anaphase, but it then seems to go away afterwards. And so here I'm just showing you two representative cells, but what we can do is we can then take this line here in multiple cells and average it to give you a profile of what's happening over time. So what you can see here is that, so this is the time of anaphase. Prior to anaphase, we've got an apparent elasticity that stays more or less constant, that increases significantly and reaches a peak around 6 minutes after the beginning of anaphase, and then it decreases over time. Whereas myosin, for example, seems to increase consistently. And this consistent increase in myosin can either be because we're recruiting more myosin to the cortex, or it could be because we've actually got the same quantity of myosin, but the cleavage furrow is contracting, so we are concentrating that on a smaller perimeter. And to try to differentiate these two, what we did is we calculated the total intensity of myosin, which is the average intensity at the furrow multiplied by the furrow perimeter. And I'll just show you the results on this graph. So what we have here is the furrow radius. So it starts and stays more or less constant even after anaphase, and then at some stage we start a linear decrease. And if we look at myosin, what you can see is that it stays more or less constant until anaphase. It increases up to about 6 minutes, which coincides roughly with the beginning of furrowing, and then it stays constant over time. So it looks like we put all of the myosin in place, furrowing starts, and then you carry on with roughly the same amount of myosin. And in contrast, if you look at actin, that stays roughly constant and then it decreases during the furrow ingression. But one thing that we found was really surprising is that if we look at the increase in myosin and we compare that to the increase in apparent elasticity, they are very different. So there's something that we're not quite understanding directly there. And essentially what it might be is that in addition to a recruitment of myosin, we've also got some reorganization of the cortex. So for example, to go, I usually do this with my hands, but with a microphone it's a bit difficult. So usually to go from an isotropic network to something that's more aligned like this. Okay, so and to really now try and link this mechanical data to the shape data, we worked in collaboration with Guillaume Salbreux and Diana Kohomskaya, and what they did was that they took our myosin data, they can segment that to get the myosin profile around the cell contour over time, and the cell volume, and they use that as an input to a mechanical simulation that's parametrized using experimental data. And what that simulation outputs is the cell shape dynamics, the velocities and cortical tensions over time. And we can take our experimental data and also segment it to get the shape evolution over time so that we can actually compare whether our simulations look realistic or not. So first, we considered that we had an isotropic increase in tension at the cortex at the cleavage furrow, and what you can see here is that at the beginning, experiment and simulation look pretty good, but actually later on it completely diverges. And what that suggested is that we really need to consider an anisotropic tension, and one source for an anisotropic tension could be this reorganization of the actin cortex to become more aligned. And so, for example, here's some images from Dylan Burnett's lab where he's labeled the rod domain of myosins in green and the motor domains in magenta. And what you can see is that these all seem to align at the cleavage furrow so that it would create an anisotropic tension. So if you put that into the model and you now say that the tension along this blue line is higher than along the red line here, you can recover an evolution of cell shape that looks somewhat more realistic. So really that means that this anisotropic tension is essential for furrowing. And one way in which you could get this type of nematic alignment is that you might have an increase in filament density that's caused by the motor activity of myosins that creates a flow of actin. And so Anne-Cécile Rehman did some of this work, and she looked at something similar, essentially in the pseudo-cleavage in C. elegans, and in this case, what they saw is that you could induce this alignment. Another possibility is that you could just have some signaling that locally generates these structures at the equator. And to try and test these two possibilities, what we did is that we treated cells with blebistatin, which is an inhibitor of myosin activity, and our reasoning was that if we treat this with increasing concentrations of blebistatin, we should be reducing the flow velocities, and that would mean that the time it takes to align all the filaments and to reach a sufficiently large density for this nematic alignment, that should become longer and longer, so that we expect to see our peak in apparent elasticity to change its timing. So when we did those experiments, what we found was that myosin inhibition reduces the absolute value of elasticity, but it doesn't actually change how much the elasticity at 6 minutes after anaphase increases relative to the elasticity before, and it didn't change the timing of that peak in elasticity. And so what that suggested is that this initial recruitment of myosin is not something that just emerges through a sort of self-sustaining phenomenon with flows, like in the pseudo-cleavage, but that really relies on integral signaling to make that structure. But one thing that does change, of course, is that the rate of furrowing changes afterwards. So it looks like we've got a first phenomenon whose timescale is independent on myosin and dependent on signaling, and a second phenomenon where we've got a shape change phase, and that completely depends on the contraction of myosins. So signaling appears important, and I told you that myosin recruitment at the equator was controlled by ROGEFs and ROGAPs, and so to determine their contribution, we depleted these with siRNA, and in particular we concentrated on MyoGEF and MPGAP. And if you do that and you plot the time evolution of apparent elasticity along the pole-to-pole axis, what you can see in green here is that when you deplete an activator of Rho you get a lower apparent elasticity, and if you deplete an inactivator of Rho you get a larger elasticity. So that all seems to be fairly consistent. If you look at the onset time of furrowing and you look at the furrowing speed, things are a little bit less obvious. So for example, if you have a more tense cortex, you've got a delayed onset and you've got a slower furrowing speed, potentially because you've now got a more contractile cortex at the poles that resists the furrow formation. So all of these data essentially show, at the very least, that localized signaling controls the tension gradients in the cortex that are necessary to give rise to shape change, and it also controls the kinetics of morphogenesis. So the shape change results from a measurable gradient in cortical mechanics. These tension gradients are controlled by pattern signaling, and the constriction rate, I mean, maybe not surprisingly, is controlled by myosin contractility. But I mean, ideally, I started all of this by saying something about signaling, and we like to do something a little bit more quantitative than just on and off. And so in an ideal world, we'd be able to predict the state of every protein in the cell, and from that we'd be able to predict mechanics. But there's lots of things that we don't know, so signaling cascades are super complicated. Here I'm showing the one for Rho. It's probably not even exhaustive. We don't know the concentration of all the proteins in there. We don't know all of the reaction constants. We're not going to manage, or at least not soon. So we need a way of just reducing all of this to something a little bit easier to handle, and we need to try and coarse grain that. So to try and tackle that, we also turn to optogenetics, and here we use a heterodimerizer pair that consists of Cry2 and CIBN. So these two proteins do not bind to one another in the dark, but when you shine blue light on them, they'll bind tightly. And in our system, CIBN is fused to GFP and it's targeted to the membrane, and Cry2 normally floats in the cytoplasm and it's fused to M-cherry and the DHPH domain of a ROGEF. So that when you shine blue light, these two proteins can interact, the DHPH domain comes into contact with Rho and it can activate it. And one of the things that's really useful about the system is that it is reversible, so if you stop shining light, then these two things separate, that goes back into the cytoplasm, and so you reduce signaling. So this is what an experiment looks like. Prior to activation, the Cry2M cherry is in the cytoplasm, and after you activate, it goes to the membrane. And so you can do image segmentation to be quantitative and determine what percentage of Cry2 you've relocalized to the cortex. And again, to look at cell mechanics, we use AFM in a slightly different way here. We've got a flat cantilever, and we just squash the cell under the cantilever. What happens when you do that is that you initially get a very high force due to viscoelastic effects, and then over time this thing relaxes to a plateau, and from this plateau the size of the cell and the height of the cell, you can calculate a cortical tension. Although I would like to say, and this is work we've been doing with Hervé Turlier, is that a lot of these formula for calculating this are actually pretty big approximations, and so really we use them to compare things rather than for giving very quantitative data. So our AFM, as I said, interface to a confocal microscope. So what we can do is we can do optogenetics while we're measuring the cortical tension. So in this experiment, we come into contact with the cell, we wait to reach a plateau, and then we activate the optogenetic system with a 200 millisecond pulse of blue light, and we can measure the change in force that occurs over time. And you can see you get an increase first as more and more Cry2 goes to the membrane, and then it decreases over time. And this is very reproducible. You can do this multiple times. You get roughly the same amplitude, roughly the same duration. So it's very nice for studying things. With the confocal microscope, we can image the different steps of signaling. So we can look at the recruitment of our DH-PH domain to the membrane. We can look at the recruitment of myosin, and we can, of course, look at the evolution of cortical tension. And if you just look at these curves, what you can see right away is that their shape is pretty similar. And so what that means is that in the particular conditions that we're in, the steps that link this biological input to a biological output, like myosin recruitment, and finally to the mechanical input, all of those steps are probably linear in the particular conditions that we're looking at. But one thing we don't really know how to do, and that we need to do before we can really get more understanding of this, is how to link our experimental input, which is the light that we shine on the cell, to the biological output inputs, which is the amount of ROGF at the membrane. And so I'll spend a little bit of time on that. Okay, so to get a better understanding of that, we can try and make a simple model of what's happening. And in this case here, we've got some inactive Cry2, which is in black, and some active Cry that gets transformed into active Cry2 by light, which is in red. This active Cry2 can then bind to CIBN, which is at the membrane. And what we know from qPCR data is that we've got about 100-fold more CIBN than red and black Cry2 here. So what we assume in modeling all of this is that Cry2 inactivates as a first order process, and as soon as it inactivates, it unbinds from CIBN. And then what you have is you've got a very simplified reaction scheme that can be solved explicitly. And you can then predict the amount of Cry2 bound to the membrane and its evolution over time, and it follows a 2 exponential function. And first what you have is you've got an increase in the amount of Cry2 at the membrane over time as it binds more CIBN, and then as the Cry2 inactivates, it will detach from the membrane. And this evolution fits our experimental data very well. So we've got a good description of what's happening for this particular for the optogenetic system. And since the temporal evolution of the different steps of the signaling cascade had very similar shapes, that suggested that the different parts of the cascade were linear, and maybe what would be happening is that they would just operate a rescaling and then a delay. And so we try to implement that in a very simplified model, and what we say here essentially is that we choose an output, here it's tension, but we say that that output is rescaled in some way compared to the input, and then there is a delay that is introduced. So what we do to try and test that model is that we first look at the Cry2 at the membrane, we fix the parameters of this double exponential, and then we don't touch them ever again. And then we try and fit the other steps of the cascade, like for example the amount of myosin or the cortical tension, by playing on this rescaling and the delay function. And what you can see is that doing that, you can fit the experimental data pretty well. And what I really like about this approach is that it gives you some type of intuition for how long it takes to set up some signaling. So the delay between the peak that you have in Cry2 and the peak that you have in myosin is about 48 seconds. So that's how long it takes you to go through activation of Rho-A, activation of Rho kinase, etc., etc. And then a little bit further down the line, our peak in tension happens with a further delay of about 10 seconds. And so again, what it says is that once you've recruited myosin for things to set themselves into motion to give rise to an increase in tension, it takes a little bit longer. So that's really, that's really very nice, but it doesn't really tell us so much about how you get these proportionality factors or how you get these delays. And so for that, we started another set of experiments where what we do is we vary the stimulus. So what we do is we fix the laser power and we use a whole set of different illumination times, and we plot the amount of Cry2 at the membrane as a function of time. And what you can see essentially is that as you increase the duration of illumination, you relocalize more and more Cry2 to the membrane up to a point where you saturate. And you can also do the exact same experiment by maintaining duration constant, but by changing the laser power. And again, as you increase the laser power, you increase the amount of Cry2 you send to the membrane up to a point when you saturate. And essentially what that says is that the amount of Cry2 that you put at the membrane is dependent upon the light energy that you put into the system. So that gives us a good description of when we would expect to be in the linear regime for the actual optogenetic system, but it doesn't really tell us about the linearity of the other parts of the cascade. And so what we need to do is we need to repeat all of these experiments, and that's what we're doing currently. We need to repeat these experiments for all of the different steps of the cascade. So for example, what we would do is we'd monitor Cry2 at the membrane with a few different, and do a few different durations of illumination. We can check in those cells to see how that changes the amount of myosin that we recruit, and you can see that myosin seems to follow about the same trend. And then we can also monitor the amount of the change in tension that we get for that. And there things are a little bit more complicated. So you do get an increase in the amplitude as you increase the duration between, say, 50 milliseconds and 100 milliseconds. But above that, what seems to happen is that you increase the duration of the peak in tension. And that's something that we're still trying to understand. And essentially, the question that it brings about is that we need to think about the whole of our signaling cascade and where the different saturations could take place. And one thing that this, when I talked about coarse graining signaling, and maybe what we can do with experiments like these is explore the use of transfer functions. So transfer functions are a thing that people use in electronics and signal processing, and essentially what they are is they're just saying, I have a black box, I don't know how it works, but all I need to know is how it transforms a signal in into a signal out. And here what we might have is the amount of Rho-Gef that we have at the membrane, that's our signal in, and our signal out is the change in mechanics, and everything that's in between is something that, as a first step, maybe we don't need to know about, and it's just the transfer function, and it just coarse grains the whole of signaling. But the main message of this particular part is that the more Cry2 you put at the membrane, the more tension you seem to get out of it. And we wanted to confirm that with orthogonal means of measurement because, AFM is great, but the throughput is low. And so we turned to a technique that was developed in Jochen Guck's lab, and that's real-time deformability cytometry. And essentially, this is a technique that relies on flow cytometry. You've got cells in suspension, you circulate them through a constriction in a microfluidic device, and through hydrodynamic forces, you get a deformation of the cells as they go through here without any contact with the channel walls. And Jochen's lab spent a lot of time developing models that essentially allow you to from an image of the cell compute an apparent elastic modulus, and they can do that on the fly, and essentially allows you to take an image, segment it, compute the elasticity, and they can measure hundreds of cells per second. So we work with them to adapt that assay to optogenetics, and what we do essentially is that we shine a blue laser on the cells before they are circulated through the constriction, and we use the fact that we've got quite a slow inactivation of our system. We've got about 10 minutes during which Cry2 and CIBN are going to separate, and so we can actually do quite a lot of measurements during this time. The other thing that's in their system is that they've also got a light sheet here, and that means that we can measure the fluorescence in Cry2 in every cell that goes through, and we can then separate our cell populations dependent upon the amount of Cry2 that they have inside. So here I've colored them in increasing order of Cry2, and what you can see is that after the optogenetic activation that happens here, the cells that show the largest increase in apparent elasticity are the ones that have the most Cry2. So again, it looks like the amount of tension increase that you get is proportional to the amount of signaling that you put in there. Okay, so you remember at the beginning, though, I told you that there were several ROGEFs that control the activity at the equator, and they all seem to regulate ρA. And so why do you have several ROGEFs? What's the point? And one possibility is that GEFs might actually differ in the mechanical response that they induced. So we compared 3 ROGEFs. So we compared P115 ROGEF RGEF1, which is in green here, EC2, which is in blue, and RGEF11, which is PDZ ROGEF, which appears in orange. And what you can see right away is that when we're looking at the amount of Cry2 relocalized, the rise time is the same, but what seems to separate them is how long it takes for them to relax and to go back to the initial state. And that translates into changes in cortical tension that are more or less long-lasting. And because the optogenetic system is exactly the same in both cases, it means that dependent on the DHPH domain, there seems to be a different strength of interaction with ρA or potentially with other ROGEFs, with other ρGTPases. And so if you now include an interaction term for this in the equations, you can then fit all of the data and essentially get some type of understanding of why we've got these differences in mechanical change. Okay, so So in summary, essentially, the myosin intensity and cortical tension change seem to be proportional to how much GEF you put at the cortex. We might be able to coarse grain signaling with things like delay functions or transfer functions, but I think we do need to try and coarse grain them in some way. And there's a number of things that we still don't understand. So in the conditions that we use, which is just one pulse of brief signaling, we don't get any saturation, but we know that there is saturation that happens in other phenomena. So under what conditions can we saturate signaling, and then what part of the signal cascade actually saturates first? And those things are really things that we that would be nice to look into. Okay, so in the last part of my talk, I'll switch to the orientation of division, and it will soon become clear why I've included this in here. So the orientation of division is really important for tissue growth, so that if you've got an isotropic orientation of division, you get an isotropic growth of the tissue. But if most cell divisions are aligned, then what you'll get is an elongation of the tissue along the axis of division. And work over many years has shown that cells can respond to external forces by reorienting their divisions. So for example, work in Mathieu Pierre's lab showed that if you plate RPE1 cells on these sort of elongated patterns and you then stretch the substrate, that will reorient the axis of division towards the axis of stretch. And similarly in zebrafish embryos, if you expose a dividing cell to a tension that's perpendicular to its division orientation, it will reorient its furrow towards the tension axis. Okay, and the common point between these two situations is that you're probably generating an inhomogeneous tension in the cortex of these dividing cells. And likely, what's happening in both cases is that you're creating higher stresses at the poles. And so we decided to try and examine how spindles respond to inhomogeneities in cortical stress. And to do that, we use the same optogenetic system as before, except that what we do now is that we just activate at the poles or in a particular region of the cell. So the first step, I'll try to convince you that we can actually get localized recruitment of Cry2 to the poles if we do that, and to get an idea that we do indeed induce an asymmetric tension, inhomogeneous tension. What we did is we measured the length of the polar axis and the equatorial axis, and we compute an aspect ratio before and after optogenetic activation. So if we do this with a control optogenetic system which doesn't have a GEF domain, then we get no change. But if we do include the GEF domain, then what we have is we do have a significant change in the aspect ratio. So it looks like local activation of GEFs will give you a local increase in tension. And we then asked what happens to spindle orientation if we suddenly increase tension. So in this movie here, we're looking at Cry2 M-Cherry with a ROGEF. You can see the metaphase plate here, and what you'll see is that surprisingly, when we activate the ROGEF at the cell poles, what happens is that the cell changes its axis of division away from these regions where we've activated. And so we can try to quantify that by measuring the angle between the axis set by the metaphase plate before we activate and the one just before anaphase. And we repeated these experiments, and we asked, well, okay, if you do a global activation and you just increase mechanics, does anything happen? And nothing happens. If you increase tension at the equator rather than at the poles, does anything happen? And nothing happens. And essentially that leaves us with a situation where it's only when you do bipolar activation that you get a reorientation of the spindle. And so we started wondering, what was happening in this case. And although I've been telling you, okay, we have an inhomogeneous cortical tension, actually what we're controlling is RhoA activity. So we also went to look at downstream effectors of RhoA. So here I'm showing you the Cry2 with the DHPH domain. And this here is the myosin in our system, and what you can see is that when you increase when you activate our optogenetic probe at the poles, you also get recruitment of myosin, and you can quantify that in these type of diagrams. And you can ask, well, what is it that's important for this rotation? You could imagine 100 things that RhoA regulates, but maybe it's not tension that's important. So what we did is we inhibited contractility either with Rho kinase or with blebistatin, and in both cases we inhibit the rotation. So it looks like contractility is important, and what's interesting as well is that blebistatin doesn't block the binding of myosin to F-actin, and so it's probably also not the cross-linking capabilities of myosin that are important. Okay. So 2 minutes, I think I'll be a little bit over 2 minutes, but I'll try and do my best. How do you go from cortical tension to spindle orientation? Well, there's a whole very well conserved spindle positioning machinery that consists of G-alpha-I, LGN, and NUMA, and this complex recruits dynein that can then exert pulling forces on astral microtubules. So we decided to have a look at that. So for example, normally pneuma is enriched at the poles of the cell, but you can inhibit that localization by inhibiting Aurora A with MLN, and that's what it looks like. And you can also block the activity of dyneins with cilibrevin, and in both cases you block the rotation of the spindle. So you really need this pulling machinery to be in place. And similarly, you can also get rid of astral microtubules, and you also need those for rotation. So one thing that could explain rotation is that actually all that you do is you just change the localization of pneuma, and that was a real concern. And so what we did is we again did experiments when we look when we have our optogenetic cells, we activate, and then we look at the localization of pneuma over time to ask if it reorients. And so if we quantify that, what we find is that we don't get any change of pneuma either at the pole or the equator, and the polar amount of pneuma seems to stay always larger than the equatorial amount. And that suggests essentially that the pulling force machinery itself is not perturbed by the activation, and what we then asked is whether or not we had a change in the force balance that's applied on the spindle. So as a first step, we checked that, we looked at what the net force spindle poles were subjected to, and for that we ablate astral microtubules at one of the cell poles, and we look at its change in position, and what you can see is that after you do that, it relaxes towards the cell center, and that would suggest that it's under net tensile forces. And after that, what we wanted to know is what the optogenetic activation did. And here, I'll just go through this scheme briefly, but what you could imagine is that, maybe optogenetic activation would give rise to an inward movement of the centrosomes, and that might be either because you reduce the microtubule pulling force, or the other thing that microtubules do is that they can push against the cortex, and maybe you increase the pushing force. Or alternatively, the centrosomes might move away from the cell center, and that would be because you've got an increase in pulling force or reduction in pushing force. And so we did experiments where we tracked the centrosomes before and after activation, and what you can just about see is that you get a little bit of an inward movement of the spindle poles, and that would suggest that we've either got a reduction in the microtubule pulling force or an increase in the pushing force. And to try and differentiate these two, what we did is that we again used this drug to remove pneuma from the cortex, so that we should no longer have any pulling forces. And in that case, what happens is that you just don't get any movement of the centrosomes anymore. And that suggests that, at least in this system, we don't have any pushing forces that are present. Okay, and I'm nearly done, but so to try and make sense of all of this, essentially we turn to numerical simulations, and what we have is we've got a spindle that is parametrized by the angle that it makes with the horizontal, and it's subjected to pulling forces at the cortex that are in a particular zone to reflect where the pneuma and dynein are located, and these pulling forces create a torque on the spindle. So we parameterize the pulling forces using the fluorescence intensity profile of pneuma, and then to model the optogenetic activation, what we do is we add a perturbation with a certain width, and that perturbation essentially reflects the sum of these two things gives you a profile of forces where you've got a reduced pulling force in this region. And we parameterize that based on the myosin profile that we change with optogenetic activation. And what that predicts is the torque exerted on the spindle as a function of its orientation. So in control conditions, if you move a little bit away from the zero position, you'll get a negative torque, so that will tend to push the spindle back to its zero position. And on the contrary, if you go to a slightly negative, it's a positive torque, so again it will push it back to zero. So zero is a stable point. But if you But if you now go to bipolar activation, we no longer have 0 as a stable position, and we've got a position that's slightly tilted away from 0. Whereas in the global conditions, 0 is still a stable position. Okay, so the model qualitatively matches our observations, and it suggests that essentially spindle reorientation happens because we've got a change in the balance of forces that are exerted on the spindle. And essentially, to try and summarize what we think is happening is that in response to an increase in cortical tension at the poles, we've got a reduction in the pulling forces, and now we've got larger pulling forces on either side of this zone, and that will tilt the spindle and bring it to a new stable position. And essentially, tissue tension would likely give rise to inhomogeneous cortical tensions in the dividing cells, and that's what the spindles respond to. Okay, so before I finish, I'd just like to thank the people who did the work. Pierre Boeck did all of the work on AFM and activating the cells. Mansi Kelkar did all of the work on spindle rotation. Ryota Sakamoto, I don't have a picture of, but he did all of the work on measuring the mechanical properties of the cleavage furrow. All of the modeling was done in collaboration with Guillaume Salbreu's lab. Martial and his team helped us with some micro-patterning that I didn't really get to talk about. Jochen Guck with the RTDC. Mathieu Copé and Léo Vallon were very kind in sharing optogenetic constructs early on in the project, and Buzz Baum's been a constant collaborator on all of this. I'd like to thank you for your attention. I'm sorry I'm a few minutes late.
We have a time for quick questions, maybe?
Great talk. So just regarding the last part on spindle orientation, so a dominant model as is the one from Manuel Thierry and Michel Bornans, and on when the cells are rounding, is that you have these actin retraction fibers. Did you look at that actually? No, we didn't look at retraction fibers. And I think that's an interesting point. In a certain respect, it's not incompatible. The retraction fibers could just be pulling on the cortex and exerting some type of tension on the cortex, which would feed into this model. But I think what's really difficult at this stage is that we don't have a good description of the stress within the cortex of cells, and as much as we would like to try and measure that with AFM, we can't measure it at the poles for reasons of geometry.
So referring to the first part of your presentation, so you kind of suggested there is an isotropy of tension at the level of the actin ring. Mm-hmm. Would it be possible to test this directly, for instance, with laser piercing?
Okay, so there is some work, I think it's by the Gerlish lab, where what they've done is they've done some laser ablations in the equatorial direction versus in the pole-to-pole direction, and what you get is you do indeed get a difference in recoil that's compatible with what we're saying.
So thanks for your talk. I was intrigued by the myosin 2 localization as spindle poles, because we have the same also in our model system. And I wanted to know your opinion. Do you think it's a reservoir of myosin, or do you think they are actually exerting forces, and could it be important for spindle orientation but also furrowing?
I have no idea. Yeah, that's possible. We could try and do that. We've never tried. I have no idea what it's doing there, but I know every single cell line I look at, and then you look at images from other people, so there's formins there as well. So if you look at the early work from Naoki Watanabe and people like that, there is formin M-dia1 at the spindle poles, and I don't know. No one wants to put their finger on it.
Very nice results. I was a bit intrigued by the shape of cells upon cytokinesis in simulations, because for me the first source of anisotropy of the tension is the fact that there might be cortical flows, which breaks the symmetry, and this is what I showed a few years back. And so does that mean that the model doesn't take into account cortical flows, or so that, of course, adding the filament orientation will add more in anisotropy.
Yeah.
But I was a little bit intrigued by the shape, which looked quite different from what
I had at the time.
From what you had initially, yeah. So in response to that, so they do have cortical flows in there, and I think they tried to parameterize the velocity of cortical flows, and that was based on other experiments that we did where we do a monopolar, we only activate on one side of the cell and we look at the shape change, and I think that that's what they used to parameterize the strength of flows, and they were trying to match the shape there. So it is matched empirically on the model and on the experimental data, but beyond that I can't tell you any more details about it.
Okay, thank you very much, Guillaume.